Hello. Hello. Hello, can you hear me? Can you please confirm whether I am audible or not? Hello, everyone. Hello. Good evening, everyone. Hello. Is the video and the audio clear? Is it okay? Can you please let me know? Can you please let me know whether the video and the audio is clear and is it clearly audible? Yes, good evening, everyone. A very good evening to all of you. Yes, sir. Loud and alive. Yes, Pawan. That should be the spirit. Clear. Very good. All right. So without wasting much time, we will directly jump into Fertility MahaMarathon part 2. So video isn't clear. Yes, I am also seeing here some buffering is happening. I don't know what is the reason. Maybe the internet is not that strong. Today there was a huge rain and storm in Delhi, So that may be the reason. Anyhow, not a problem. Technicalities तो होते रहेंगे. Wait, I'll ask. Screen lag हो रहा है बोल रहा है. Connection poor क्यों है? Like बारिश हुआ इसलिए क्या? Huh? Hello, can you hear me? Yes, the connection is poor. I can also see that, but we are trying to make that right. There has been a storm today in Delhi, so that might be the reason. We are trying to make that correct, so please wait for a few more time. अभी good हुआ. Hello everyone. Can you give me the feedback now? Is it clear? Is it coming okay? We tried to mend it. Hello, can you see clearly? Your audio and video is okay? Correct आ रहा है? Can you please let me know in the comments? I think आ रहा है. ठीक है. Thank you. Okay, Pawan, I think now we are good to go. Is everything is correct? Video, audio, everything is going good, huh? Yes, so now I think there will be no more problem and we will dive deep into fertility. So in the last class, we have covered fertility until we have covered mineral nutrition, we have covered nitrogen, phosphorus, potassium, and micronutrients as well, right? We have covered all those. So in the last class, I think 1 or 2 portions were left out of which one thing I commented in that video, that is leg hemoglobin. So you have to read about leg hemoglobin while reading about nitrogen fixation and that. So leg hemoglobin का function क्या है, leg hemoglobin कैसे बनता है, and what is the exact function of leg hemoglobin in relation to both the bacteroid and the plant. All right, this is the one part that we left out, and another part that we left out in mineral nutrition was the theories of ion uptake, right? We talked until the theories of ion movement, diffusion, mass flow, and root interception, but theories of ion uptake, we have left it out because if you analyze the question, it is very rare, it does not come, but still today we will cover one of that theory, that is the carrier hypothesis. We will cover very small, and also another वहाँ से क्या क्या question आ सकता है वो भी मैं बता दूंगा. All right, so I think everything is nice, we are very good to go. So let's jump right into it. So today's topic is definitely fertilizers and manures. So these parts are also, it might feel a bit boring, it might feel a bit के अरे सर यहाँ पे तो बहुत ज्यादा याद रखना पड़ता है, fine, but you have to remember यहाँ से question भी बहुत ज्यादा आता है, like the number of questions that come from these portions are exceptionally high. So you can expect at least, yesterday, like in the last class I was telling, almost 25 to 30 questions are coming from fertility every year, if you take the whole fertility as a one whole. So we cannot leave anything, right? So manures, I think all of you know, there is nothing new to say. Manures are organic material. I have just highlighted the keywords for you. There is nothing to worry about. Just see the keywords, just keep them in your mind, and we will be good to go. So manures, definitely सबसे पहले जो आना चाहिए, that is organic material. It is of course organic material that will be plant, animal or human origin. Animal or human तो दोनों same ही आता है, but still for giving human some high level, I have mentioned it अलग से. Containing, here is another keyword for you, small amount of plant nutrients. All right, these are also low analysis organic fertilizer. This is another keyword, that is low analysis. It has, what is the meaning of low analysis? That is, it has lesser nutrient concentration. It has lesser nutrient concentration compared to fertilizers. We all know nutrients are present in complex organic forms, because manure जो है उसमें the nutrient is not available as such, it is not available very fast, very rapid. That's why nutrients are present in complex organic forms, because organic matter है तो mostly जो nitrogen है that will be associated with the protein, right? Then जो phosphorus है that will be associated with the other compounds, the inositol phosphates, the nucleic acids and everything. All right, so please remember, nutrients are present in complex organic forms. Nutrients are present in complex organic forms. And then finally, it maintains optimum physical, chemical and biological properties of the soil. This is like the main thing of using organic manures. All right, so the difference between manures and fertilizers, this I don't think I need to tell to any JRF aspirant who is giving exam just 20-30 days away. So still you can see, it is decomposed organic matter, it is chemical formulation, it is formed from natural materials like plant animal residues, synthetic formed from chemical salts, mineral content, small quantity, large quantity, then specificity, not nutrient specific, nutrient specific. This one is very important. You cannot make manures nutrient specific. Manures are organic materials. These are plant, animal and human origin. So you cannot make it nutrient specific. सिर्फ nitrogen manure हम नहीं बना सकते, right? Organic matter adds organic matter to the soil. Very common sense. Quantity required. This is also a major problem of using organic manures. It is required in bulk. It is required in huge quantity. That is the reason it becomes sometimes economically not feasible. बहुत ज़्यादा पैसा लगता है for its transport. Yes, if you are transporting 5 tons, 10 tons of organic manure, it will require lots of transportation and lots of expenses. That is another reason why organic manure cannot be used that easily as we can use chemical fertilizers. The nutrient availability is moderate and released very slowly, because all nutrients are present in chemical combinations. पहले उसको तोड़ना होगा and then only it will release. So definitely please remember, this is the key word, it will always be slow. Then manure is bulky in nature, difficult to transport to longer distance, as I was telling. Excess manure is not that harmful. You can add as much manure as you want because it will improve the soil physical property. Yes, we know soil organic matter is a very huge indicator of soil health also, and the cost is expensive because this is very expensive. And the same is for fertilizers there. You can read this PPT if you want. So manures का अगर हम classification करेंगे, if we classify manures, organic manure can be classified into bulky and concentrated. Concentrated can be classified into plant origin and animal origin. I don't think there is any doubt in this. If there is any doubt in this, you can ask. I don't think there should be any doubt in this. Bulky, you all know, lesser concentration of nutrients, and it has huge bulky nature. The volume is very high, but in the case of concentrated, the nutrition quantity is comparatively more than the bulky, and it is concentrated in nature, like the volume is less. So this is the detailed classification of organic manure. If you see, this has been made using AI, you can see. So among bulky organic manures, the main ones that will come: FIM, compost, sewage, sludge, vermicompost, all this. All right. Then in concentrated, you know, animal oil cakes of plant origin and animal origin. In the oil cakes, there will be edible oil cake, non-edible oil cake, and animal origin is blood meal, bone meal, fish manure, and guano. Then for green manures, you all know there are two types of green manure. One is in situ green manuring, another is called green leaf manuring, also called as ex situ green manuring. All right. ये सब बाद में भी आएगा. We will discuss it there. This is just a overall classification. You can see. So moving on, this is the percentage of bulky organic manure which is having what percent of N, P2O5 and K2O. I will be very honest with you. I don't think this one is much required from your exam point of view because there is very less chance they will ask poultry litter में कितना nitrogen है बताओ. So you can read it from treatise. You can go through it once, but remembering all this data, I don't think that is totally feasible. All right. Moving on. Now this table I have made it alag se for you because this is important. From here you can get question. But again, that question will not be like kitna hai batao. It will not be like that. You can see this one is made here as bold. Specifically, this one is made here as bold. Can anyone say why this is made bold here? What is the reason? What is the reason of making it bold? And question aayega, so most probably it will come from this row only. What is so special about it? Can anyone tell? That is very simple actually. I'm just trying to interact with you so that I also don't feel bored. Yes, Shreyoshi, you are absolutely correct. You can see highest 10. Among all the listed here, this has got the highest 10. So in some PYQs, I have seen this. Okay, which one has the highest percentage N? So you have to remember it is decorticated safflower cake. The other also, you can remember, this is first, this is second, 1 and 2 you can remember, but remembering all the values, that is not at all required, and please do not put your pressure in the brain remembering all these things. ये ना हो कि तुम ये याद रखो और फिर जाके theories of ion exchange भूल जाओ. That will be very sad, right? So again, in the concentrated organic manure of plant origin which are non-edible, the list that was in the previous slide, it was edible oil cake. This time it is non-edible oil cake. In the non-edible oil cake, you can see again I have highlighted the neem specifically just because again the same reason, that is the highest percentage of nitrogen. All right. All right. So ये सब तो हो गया. Then concentrated organic manure, animal origin. Here, from here also you can expect questions. That is, blood meal has got the highest nitrogen. But here there is another more important question which has been added in the bracket. All right. So in this slide, this is a more important question rather than which has got the highest nitrogen. Why so? Because you can see these are the differences between raw bone meal and steamed bone meal. Yes. In raw bone meal and steamed bone meal, nitrogen content is not important. What is important is this column. Here you can see the phosphorus percentage P2O5 here is 20. In case of steamed, it is 25. So when you are steaming the bone meal, the phosphorus content is increasing. Can anyone tell why is that happening? That is very basic common sense only. But can you tell why is that happening? Why is the phosphorus increasing in case of steaming the bone meal? In case of steaming the bone meal, the P2O5 content is increasing. Why? Because with the application of steam, we are denaturing the bonds, right? The phosphate that is present in different chemical combinations. Aishwarya, I will tell that, don't worry. So whichever phosphate is present in different chemical composition, when you are applying steam, it is denaturing those molecules and those compounds and is making the phosphorus more available, because this is normal common sense only, right? This is the classic case of mineralization only. You are advancing the process of mineralization. जो बहुत सारे combination में है, उसको heat देके, steam देके, we are trying to break that so that the phosphate becomes easily available, right? And here, which is very important, is this what have been included in the bracket, 8% citrate soluble, and in this 16% citrate soluble. What is the meaning of citrate soluble? Aishwarya is asking. See, phosphate fertilizers, phosphatic fertilizers. Nikhil sir, this lecture is also useful for agronomy. Yes, definitely. Why not? Agronomy mein to fertility is the biggest portion actually. If you see in RS Meena agronomy facts for competition, you will see soil and plant nutrients. That chapter is huge. Definitely, it is useful. You can see. Yes. So while we are classifying phosphatic fertilizers, if you remember, Aishwarya, that there are two ways, actually three ways. One is called water soluble, one is called citrate soluble, and another is called citrate insoluble. So phosphatic fertilizers are classified on the basis of these three parameters. So what is water soluble? Water soluble ka matlab hai it is soluble in water. Then citrate soluble means it is not soluble in water, but it is soluble in citric acid. That citric acid ka jo concentration tha, that is also important, but at this point of time I am not remembering. If any one of you can help me, I will be very happy. What is that concentration of the citrate which we use? So if that is soluble in citrate, that is called citrate soluble phosphate, and the other more hard type of phosphate like phosphate rocks, those are not soluble in water, those are not soluble in citrate. That is called citrate insoluble phosphate. So here you can see in case of raw bone meal, it is only 8% citrate soluble phosphate, whereas when you are applying steamed bone meal has 16% citrate soluble phosphate. So the citrate soluble phosphate is almost increasing double, right? It is becoming double. That is why steamed bone meal is always better to use than raw bone meal to get quicker nutrient release and more nutrient release as well. Because यहाँ पे 8% citrate soluble है, rest other is citrate insoluble. So making that mineralizable, making that phosphate available for plants, it will take a long time, right? But in this case where there is 16% citrate soluble, that can be available to the plants in a shorter amount of time. All right. Is it clear, Aishwarya? Can you let me know? Still, if there is any other question, you can ask me, definitely. Can you tell me, Aishwarya, is it clear? Yes, Pawan, you are also correct in a point. Other nutrients are volatile, such as nitrogen, but in case of phosphorus, there is no volatility. All right, Aishwarya, clear hai, bahut badhiya. Moving on. So, we are coming to farmyard manure. Farmyard manure is a bulky organic manure. Just we were discussing, right? In that classification, it is a bulky organic manure. That means it is highly voluminous, but the nutrient concentration is comparatively lower. Okay. So here I have written only those properties which are extremely important for you. ठीक है? So यहाँ पे जितना लिखा है, you have to remember. There is no other way, right? So let's see what is written. It is the decomposed product of dung, urine, litter, and leftover fodder fed to the cattle. FOM becomes ready for use in 4 to 6 months. This 0.5% nitrogen, 0.2% P2O5 and 0.5% K2O. This has been taken from old ISSS. the chapter written by Dr. Narayan Swamy and Dr. D.R. Vishwas. This has been taken from there, and these values I have seen in many questions, in many PYQs, this always comes. Please remember these values as it is. 0.5, 0.2, 0.5. All right. Then 30%, then 60 to 70% P2O5 and 75% K2O is available to the crop in first year of application, and rest of the nutrients is available in the subsequent year. This is not true for nitrogen because nitrogen gets very easily lost. All right. Phosphorus, P2O5 and K2O, these have the residual effect, as you all know. So इसमें 60 to 70% P2O5 will be available in the first cycle, then the rest will be available in the next cycle. Same for potassium. But in case of nitrogen, only 30% nitrogen will be available, rest nitrogen will be lost. That is called reactive nitrogen loss. Okay. So यहाँ पे समझाने का और कुछ है नहीं. I think all of you know how FOM is prepared. Preparation of FOM, I don't think there is any need to tell that. Yes, I think you all know. In the cattle shed, you have to put those plant litters in the cattle shed where the urine and the dung will be collected. Then that urine soaked and dung soaked litters, those will be taken and put in the trench, and we will make FOM, right? So उसमें बोलने का कुछ नहीं है. This is very, very important. This is very important. Factors affecting the quality of FOM. This is very important. You should remember all of these factors and especially the logic behind these factors. What is the reason? Source of excreta. This is kind of common sense. If the source of excreta is good, animal, young and healthy animal will provide a better FOM quality. What is your view in this? Can anyone tell me? Can you please tell me age and condition of animal, how is it related to the quality of the FOM? Whether an old and sick animal will provide better quality FOM than a young and healthy animal? If you can please write. Anyone? All right, you keep on writing. I think someone of you will definitely write. Otherwise, I will tell. Moving on to the next function. The function of animal. Function of animal is also important. I guess old animal excreta will be more in nutrients. Yes, इसको guess करने का क्या है? That is clearly written in old ISSS. Why are you still guessing? JRF is just few days away. It is only 15, 16, 20 days max. So you should not guess now. You should be confident. No, Aishwarya, exactly. You are wrong. Aishwarya, you are wrong. Nikhila, you are wrong. Totally wrong. Vipin, again you are wrong. कोई बात नहीं, समझा देंगे. No issues. That's why you all are here. We are all learning, right? When the animal is young and healthy, जब हम लोग young and healthy होते हैं, तो whatever food we are taking, we are assimilating also, right? We are eating chicken, we are eating vegetables, we are eating everything. Whatever we are eating, we are assimilating the nutrients that is used for our body building. Here we are only interested about the excreta. Please understand that. We are not using the whole animal as the manure. The animal is not taken here, only the excreta, right? So young and healthy animal, it will take more of its nutrients for its own body building, right? Because usko muscle banana hai, young hai, healthy condition mein hai, it will absorb all the nutrients. So the excreta that will be released by them, that will contain less nutrients. So that is not good as a FOM material. Whereas when the animal is old and sick, it is less efficient in absorbing nutrients. When we also get old, we also have the same problem, right? Whatever we eat, we are not able to assimilate that in our muscles, in our bodybuilding. So उसमें क्या होगा? Higher proportion of nutrients, the N, P and K, that will be released into the excreta. So from FOM point of view, we are only interested with the excreta and we are not interested with the animal. Animal से हमें क्या मतलब? So excreta जो आ रहा है, the excreta that is coming from old and sick animal, that will contain higher percentage of nutrients. So it is a better dung, it is a better raw material for FOM. Am I clear? Aishwarya, Nikhila, Vipin, all three of you who have written that, is it okay? Or you need any other explanation? This is clearly written in old ISSS. If you go and read it today, just beside that FOM portion where these things are written, factors affecting the quality of FOM, this has been clearly and very beautifully, they have explained this portion, all right? You can read from there also. Baki, you should have understood by now, I think. So the function of animal is also the same. Function of animal also, you think from the same point of view. Yes, function of animal, if an animal is busy in huge amount of work, that is, it is doing, say it is being used in plowing the field, it is used for what they call those tongas, you know, jo bail gadi hota hai. It is used for taking those bail gadis out, and it is used for plowing, and that animal is engaged in huge amount of physical activities and physical labor. In that case, again the same system, that will absorb more number of nutrients, more proportion of nutrients. So in that case, an animal which is functioning very heavily, very efficiently, uska jo dung hoga, that will contain less nutrients, whereas an animal that is sitting idle, that is sitting isolated, bodybuilding mein kuch nutrient nahi ja raha hai, that will give more nutritious dung that can be used as FOM. So in this also, it will be the similar as that of age and condition of animal. All right. Yes, Pawan, absolutely correct. Non-functioning animals dung contain more nutrients. Same logic. Same logic. जो कुछ काम नहीं कर रहा है, पूरा दिन बैठा हुआ है, it's very natural, it will not use the nutrients that it is taking. All right. Then manner of storage and condition of cattle shed, this is also very important. The way you are storing your FOM, that will definitely decide the nutrient quantity, because if the storage condition is not good, everything will be lost. Then the nature of the litter, that is also there. Then loss of nutrients during collection of preparation and storage. There are two major pathways. One is leaching, one is volatilization, and very sorry to say that both of these pathways are linked to nitrogen only. All right, nitrogen ka hi loss hota hai jo hota hai, because nitrogen, once, see, when nitrogen is in the atmosphere in inert condition, in the triple bonded condition, in that case, yes, in that case, the nitrogen is non-reactive. Lekin once that nitrogen is fixed, that becomes very highly reactive. एक बार वो ट्रिपल बॉन्ड तोड़ के उसको हमने कुछ और बना लिया, then that nitrogen is very highly reactive. Everywhere in field also, if you see, even after decades and decades of research, the nitrogen use efficiency till today can go maximum 40%. Maximum, I am telling 40%. In lowland rice, it may be 30, 35. In upland crops, 40 is very rare. It will be around 35, 36 like that. Okay. So, iska kya matlab hai? When we are applying 100 kg of nitrogen, we are only getting 30. Matlab 30 plus 70 equal to 100. 70 kg nitrogen is getting wasted whenever you are applying that in field, because it is so highly reactive and the loss pathways are so huge. It will get volatilized as ammonia. Denitrification, nitrification ki wajah se it will get emitted as N2O. Then it will get leached down as nitrate and ammonium. Ammonium ban ke wo fix ho jayega. So this loss is so huge. So for nitrogen only, you can see these two pathways are there for nitrogen. Nitrogen ka leaching hoga, nitrogen ka volatilization hoga. So these are the mechanisms of loss. Leaching, you all know, the nutrient will go in the groundwater or will go into the contaminated, like it will go into the groundwater and it will be lost. And volatilization is nothing but due to the pH, it is a purely chemical reaction. Volatilization will be like ammonium is there. In this reaction, this ammonia will be volatilized. All right. Then finally, the improved methods of handling are biogas compost plant, preparation of FOM by trench method, use of chemical preservatives like gypsum and SSP. All these are very nicely written in old ISSS. एक बार please इसको पढ़ लेना. This is very, very important. That's why I spent so much time on this one slide only. FOM is very important. It is written in old ISSS very nicely. Please read that part. All right, then moving on to compost. So composting is a process of converting organic residue of plant and animal origin into organic manure, which is rich in humus, plant nutrients by a variety of microorganisms in a warm, moist, aerobic or anaerobic environment. It is a biological process. Well decomposed compost should have a neutral pH, definitely. CN ratio less than 20:1 and contain more than 16% carbon, 0.5 N, 0.5 P2O5 and 1% K2O. एक बात हमेशा याद रखना, the basic difference between compost and FOM is nothing but the raw materials that are used. In the farmyard manure, it will only be the crop litter, जो crop का जो पत्ते and other things है, that cow and the dung of the cattle and the urine. That will be the only raw material. In case of compost, the raw material can be a variety of things. It can be night soil, it can be vegetable peels, it can be whatever. But in FOM, the raw material is bounded by some constraints. That is, it has to be from the farm only, and mostly it is leaves and litters of plants, dung of cattle and urine. All right. Here you can see a pictorial description. That is also nice. Moving on. So composting. In composting, since it is very similar to FOM, do not put that much emphasis on the preparation and all. But what is important in composting is this slide. This slide is VVVI, very, very, very important, you know. All the classification of compost. You can definitely expect question from this. I have seen question from this in PYQs, in JRF, in SRF. So please be very thorough. These are very easy. There is no rocket science. एक बार पढ़ने से ही समझ में आ जाता है. So based on the substrate used, compost can be divided into rural and urban. Rural is use of farm waste, weed, cattle dung, urine, litters. That is whatever is available rurally. In case of urban, use of night soil. Night soil, you also might have seen the cars, those tankers will come for collection of night soil from the septic tanks of the households, right? So in urban compost, this is the major material. Then municipal sweeping, definitely शहर है, municipality है, उसका जो waste है, that one. Wood ash, waste from fruit and vegetable market. So based on substrate used, it is rural and urban compost. Very nice. Based on oxygen use, that is extreme common sense. It is aerobic composting and anaerobic composting, you know, as usual. In case of aerobic composting, absence of foul odor, rapid, optimum moisture, high temperature. These are all the characteristics of aerobic respiration, right? Aerobic respiration, जहाँ पे oxygen होगा, वहाँ पे absence of foul odor, it will be rapid, optimum moisture, high temperature. The compost will be ready in just 4 to 5 months. But in case of anaerobic, it takes a longer time. In anaerobic composting, low temperature, foul odor, slow rate, it will take a longer time to get prepared. All right. Then based on temperature, all these are taken from old ISSS only. Line by line, these are taken from old ISSS, I am telling you. This is no ChatGPT, no Google, nothing. Simple old ISSS. Based on temperature, mesophilic, thermophilic. Mesophilic composting, moderate temperature of 15 to 50, whereas thermophilic, 45 to 65. Based on technological approach, this one is most important among all these classifications. This one is most important, you know, because this is not usually known to us. Based on technological approach, open or window composting and mechanical composting. In open composting, the entire process is carried out in open in elongated windrows, right? And in case of mechanical composting, it is carried in enclosed digester. This is also there in old ISSS. Please ek baar dekh lena. If you want to see the photos of windrows, that also you can search in Google. You can get it. But remember, please remember this. It's very important. Then again, factors affecting composting. These are very easy to understand. Why not? Moisture content, we all know moisture is the essential part for activity of microbes. Moisture agar nahi hoga, composting will be definitely slow because moisture is needed for the microbes to work. Then temperature, again same. In the last slide only you saw temperature ke basis pe composting ka reaction rates will be different, and also temperature is required for altering the reaction rate, because we know there is a law, right? With every 10-degree rise in temperature, the reaction rate almost becomes double, right? So mostly the reaction rate will increase with temperature, but until a point, if you go up to 90, 100, definitely it will not increase. Then pH is also optimum. pH का curve हमेशा याद रखना. Any biochemical reaction, any reaction where there is microbes involved, pH का curve is always like this. All right. This is a typical example of bell-shaped curve. इसका क्या मतलब है? इसका यही मतलब है. pH इधर है, rate of reaction इधर है. इस ग्राफ का यही मतलब है. At a optimum pH, the rate will be the highest. उससे ज्यादा pH अगर है, if it is alkaline, more alkaline, the reaction rate will go down. If it is more acidic, then also the reaction rate will go down. So where there is microbes involved, majorly, where there is microbes involved and there is rate and pH relation, always remember it will be like this. Then CN ratio also we all know. If the CN ratio is very huge, the CN ratio is say 100:1. It will take a lot of time. To make it as final product of compost. If the CN ratio is very narrow, say 20:1, 30:1, in that case, the rate of composting will be high. अब ये क्यों है? I would expect all of you know that. If any one of you don't know that and you want me to tell, you can say it in the live chat and I will explain that. Then aeration is again the same thing. In the last slide only we saw aerobic and anaerobic composting. Aeration के हिसाब से aerobic composting का rate and anaerobic composting का rate depend करेगा. And finally the inoculants. If we are applying some microbial inoculants, which is also called augmentation of the inoculants, that is simple addition of some inoculants in the composting, then we can increase the rate of reaction. As simple as that. Then this one is also very important, very important. This has also been taken from old ICARS only. Old ICARS is our Bible, it is our Gita, it is our everything. So old ICARS to bhai padhna hi hai. So indoor method of composting, I have only taken the points you need to remember, right? You need to remember. So only those points have been taken. So here you can see materials required are plant residue, dung, urine soaked earth, wood ash and water. Catalytic material is animal dung. Site selected for compost pit should be near cattle shed and water source, because cattle shed will supply the raw material and water source will supply the moisture, right? ये PDF को देख के यहाँ से ऐसे पढ़ लेना. I am assuring you this will be more than enough. बार बार जाके you don't have to read line by line. This will be everything. Then trench size should be this, or it can be altered based on your availability. Continued layering, then covering the top with thin layer of soil. The filling is completed within this much. Watering 68 to 80% moisture. This is an important part. This bullet is important. Please read that. 3 turnings are required at 15, 30 and 60 days of composting. Since this is an aerobic method of composting, ये बाद में है. We will again discuss. This I have put it in a table for you to remember it more clearly. All right. Compost is ready in 4 to 5 months. Since this is aerobic composting, we'll come to that. I promise you. Don't worry. Then second is Bangalore method of composting. This, the first line is very important. It is an aerobic process. Definitely it is an aerobic process. ये क्वेश्चन बहुत जगह में आता है and people get very confused. Please don't get confused. इसका आंसर सिर्फ और सिर्फ anaerobic ही होगा. The only answer of this will be anaerobic only. कोई aerobic anaerobic का mixture, some part aerobic, some part anaerobic, whole part aerobic, nothing. The total process of Bangalore method of composting is anaerobic only. ठीक है? Remember, it is not WAD, it is WAD, okay? वो सब आ रहा है, don't worry. So here also Bangalore method, anaerobic process is very important. This I have seen in many PYQs, many PYQs, okay? Then pits are filled, this ये तो process है, you can read. Organic N compound gradually becomes soluble, fine. Loss of ammonia is minimum, because in high concentration of CO2, it is stable. Here you can see, because this is anaerobic, it is taking more time. Just now we were discussing 2-3 slides back. If it is aerobic, it will be very quick. If it is anaerobic, the time required will be much high. Plastering of pit prevent fly nuisance, control foul smell and kills pathogenic microbes. Recovery percentage of finished product is higher. The cost of preparation is less. Because why the cost of preparation is less? Since this is anaerobic process, you do not need to turn it. In the indoor method we saw 3 turnings are required. 3 turning means 3 turning के लिए you need labors 3 times. and labor is nowadays the most expensive input. All right. So the cost of preparation of this is less because there is no labor requirement as such. This one is sadly not in old ISSS, and but you have to read because this is very important. This, I think, wherever you have studied, whichever UG you are studying in college, this will definitely be there. This method of composting will definitely be there. Yes. So this is called the NADEP method of composting, right? इसका ये जो specifications है, इसको please देख लेना. 10 feet, 6 feet, 3 feet size, 30 kg manure is prepared with 1 kg dung. Wall of bricks with gap. This with gap is the keyword, or this is the characteristic feature of this composting method. Here you can see, I think all of you have seen this. This photo is taken from any composting pit only. All of you have seen this. ऐसा एक well type का रहता है, and in this you can see there are gaps, right? So these gaps are given for aeration and removal of excess moisture. If the moisture is excess, that water will go out from these gaps. If you need aeration, air or oxygen will enter from these gaps, right? So it is usually done with a thatched roof. Here you can see एक roof रखना पड़ता है. What is the need for roof in composting pits? Both in case of composting and farmyard manure preparation, it is better to have a roof. Can you tell me why? What is the reason? Can you tell me what may be the reason for that? Why a roof is needed? To reduce volatilization loss and prevent excess water in rainy days. Yes, absolutely correct. Yes, Aishwarya, एक तो है protect from rain and also to protect from sun as well. Sun और rain दोनों से बचाना है ना? Because of rain can affect the composting procedure, and also excessive sunlight, it can lead to lack of moisture and it can lead to volatilization losses. All right, very good. Here, thatched roof, then periodical application of water. I don't know how many of you have done this composting practical in your UG, but I myself have done it, you know. So periodic application of water, all these things are very known to me already. So, but you have to read, these specifications are very important. And then 32.5 tons of manure are prepared from single pit in 90 to 120 days. Here also you can see it is only 3 to 4 months. So wherever there is aerobic method is coming, you can see the time required for final preparation of compost is less, because aerobic reactions are faster. When oxygen is involved, the reaction rate is faster and the compost is prepared within 3 to 4 months. But in case of Bangalore method, which is anaerobic method, it will take almost, what was 6 to 8 months, right? It will take 6 to 8 months. ठीक है, इसको याद रखना. Fine. Then finally we are coming to the table that I was telling you. This table, if you remember, I don't think you need to read from anywhere else, because this is the table which is consisting of all the questions, right? So first box is always introduction. This is the intro. So it is developed by Albert Howard and Y.D. Ward. Pawan was writing Ward. It is not Ward. It is Ward, right? Yaad rakhna. Spelling. Sometimes NTA might play with spelling also and play with our emotions also, like it always used to do. So developed by Albert Howard and Y.D. Ward. यहाँ पे Albert Howard का नाम आ ही गया, तो can anyone tell Albert Howard is regarded as the father of what? Albert Howard is regarded as the father of what? This is a PYQ I am asking. JRF 2024. I specifically remember JRF 2024 physical science exam. This question came. Anyhow, लिख दो हाँ, बढ़िया, father of organic farming, very nice. So it was developed by Albert Howard and Y.D. Ward at the Institute of Plant Industry in Indore, India. Since it was developed in Indore, that is why it is called Indore method of composting, right? Organic farming father, modern organic farming, exactly. The most appropriate answer given here is by Vipin Yadav. Yes, it is modern organic farming. Modern organic farming का father है ना? ठीक है. बाकी you guys are also correct, not a problem. But modern organic farming zaruri hai because organic farming is an age-old practice. Yes, organic farming used to be taken place in the ancient India, ancient all the countries, right? Before Christ days, like when there was nothing. During that time also organic farming was there. So Albert Howard to tab the ही नहीं ना. So Albert Howard is the father of modern organic farming. Fine. Moving on. It was developed between 1924 and 1931. ये 1924 को याद रखना. This also might come. This is also taken from old ISSS, all these lines. So here, this is written very crisp way. If you read this PPT, everything will be perfect from the time of Baba Adam. Yes, exactly. So इंडोर मेथड इज एरोबिक. एरोबिक के साथ साथ क्या आता है फ्री में? एरोबिक जब भी है तब भी टर्निंग्स रिक्वायर्ड है, एंड टर्निंग्स रिक्वायर्ड है मतलब इट विल रिक्वायर लेबर एंड इट विल बी एक्सपेंसिव. ऑलराइट, ये सारे चीज कोरिलेट करके रख लेना. कोरिलेट ऑल दिस थिंग्स एंड इट विल बी वेरी इजी व्हेन यू आर रीडिंग, राइट? देन एरोबिक का मतलब है एरोबिक माइक्रो ऑर्गेनिजम्स आर नीडेड. Then it is high temperature, optimum moisture, rapid process 4 to 5 months because it is aerobic. Again, there is a correlation thing. Because it is aerobic, it is taking rapid, it is 4 to 5 months. Because it is aerobic, it is taking turnings, and turnings require labor, and that is making it expensive process. All right, more labor cost. Fine, I think we are done with indoor method. Indoor method you can understand very well. Coming to Bangalore method, developed by C.N. Acharya. This question I asked while I was covering the most expected series. C.N. Acharya was associated with which institute? Indian Institute of Science, Bangalore, right? The #1 institute of India, Indian Institute of Science. 1939, इसको तो याद रखना ही है. This is very important. 1939 is very important. All right. Then again coming to this anaerobic. Anaerobic के साथ साथ no need of turning. No need of turning मतलब low labor cost, right? Correlate करते हैं. Anaerobic microbes, definitely. Low temperature, see, high temperature is also related to aerobic, low temperature related to anaerobic. Then slow process 6 to 8 months. Again, this is because it is anaerobic. Then low labor cost, labor required only for digging and filling the pits, and labor for turnings is not required. So 3 turning का 3 time का labor cost you are saving. Fine? अरे इतना भी correlate नहीं करना है भाई. Jenny and Overstreet. Okay, very good. That is also 1939. Yes, we have read that in the last class only. Very good. Then NADEP composting developed by NADEP Kaka. Here I have done some shortcut for you. NADEP Kaka. Can anyone tell me what is the full name of NADEP Kaka? What is the full name of NADEP Kaka from Maharashtra? What was his full name? थोड़ा सा बड़ा है, I understand, but I would expect you guys to know. ये तो जानना ही चाहिए भाई. Narayan Devrao Pandhari Pandey. Perfect. His full name was Narayan Devrao Pandhari Pandey. From that, NADEP has come. He was a Gandhian. ये भी question में आता है. He was a follower of Mahatma Gandhi ji. So he was a Gandhian. Fine. This is also aerobic. क्यों aerobic है? Because just in the last slide we saw यहाँ पे brick wall में gaps है. So these gaps makes it aerobic. Aerobic है. Optimum moisture है. Rapid process है. All these are correlated. This low temperature, here it is low temperature because this is not the heap method. Until and unless you are putting it in a heap, there will be not much development of temperature. All right. So coming to enriched compost, yes, Shreyoshi, very good. Yes, Vipin, Raman, Pavan, everyone, very good. It is very nice to see the Josh being so high. I'm liking it. So what is enriched compost? Enriched compost, so तो नाम से ही पता चल रहा है इसमें enrichment हुआ है. When we are enriched, some people gets enriched by reading books. It is just like that. Enriched का मतलब क्या है? You are able to perform in a higher level than you originally were, right? So enriched compost is ordinary compost like FYM vermicompost or crop residue compost that is fortified. Fortified, I don't think any agricultural student needs introduction. What is fortification? Addition of something with additional nutrients or beneficial microbes to improve its nutrient content and efficiency. यहाँ पे जो definition है, you have to remember these two keywords very, very strongly. Because we have a mindset of taking it as addition of nutrients only, but enrichment can be both addition of nutrients as well as addition of beneficial microbes. सिर्फ nutrients ही डालना है ऐसा नहीं है. Microbes डालने से क्या होगा? Again the correlation. Beneficial microbes will do mineralization. It will enhance the process of mineralization. That in turn will release the nutrients. That in turn will release the nutrients fast. So that will release the nutrient fast. So either we have to release the nutrient fast, or we have to enhance the nutrition quality or the nutrition percentage of the compost. So it can be both. It can be both nutrient and microbes. All right. Then it is mainly used to increase the nutrient availability. Here only we can see that. Improve soil fertility and structure, enhance microbial activity, reduce chemical fertilizer use. Types of enrichment can be nutrient, biofertilizer, organic enrichment, integrated enrichment. You can add lots of things to make it enriched. जो तुम्हारा मन में आए, you can add simply. But always you have to remember the economics, because ऐसा एक product बना दिया जिसका price is huge, उसका कोई मतलब नहीं है ना? Then limitation of bulky organic manure. What are the limitations of bulky organic manure? This type of question can definitely come in statement type of question. They will give you 4 statements and ask which of the following are the limitation. So these questions are generally solvable by common sense only. If you read the English statement देख के पता चल जाएगा एक wrong है या नहीं है. Still, I have listed them for you. Limited availability of organic manure. High cost of preparation, high cost, everything is high cost. That is the major problem of organic. Storage, transport, application of FOM or compost, low nutrient content. To overcome these problems, enriched compost is prepared. Low nutrient content is also another major problem, because low nutrient content in a country of population 1.5 billion, low nutrient content fertilizer se simply nahi chalega, because we are not prepared only. If overnight, from tonight only we stop chemical fertilizers, within the next year only we will have famines, right? Because in a country of 1.5 billion population, you cannot maintain the food security only with organic manure, right? So various enrichment can be enrichment with nitrogen, phosphorus, potassium, all NPK together, enrichment with bioinoculants. Advantages: more concentrated, increased fertilizer efficiency, decreased losses, lesser problem in handling, efficient utilization of low grade materials. These are very easy. Then we have come to vermicompost. Vermicompost we all know. Vermi का मतलब है using of right earthworms. यहाँ पे एक soil biology का question. Earthworms are also called as ecosystem dash. Earthworms are also called as what? Ecosystem. Yes, I know that is very easy for you guys. इसमें कुछ नहीं है. So vermicomposting is a composting process aided by earthworms. Very easy. इतना ही याद रखना है. इसमें कुछ ज़्यादा याद रखना नहीं है. It is prepared in 8 weeks or within 3 months. No need of turning, because vermicompost में earthworms will do that work for you. Rate of application is at 5 tons per hectare. Commonly used species. This is very, 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 very important. I cannot stress enough. You have to remember, this can come in a variety of ways. They will give you 5 names and ask which 4 are used. They will give you 3 out of this and ask which 3 are used. They will give you all 4 and ask to select A, B, C, D. They might give in statement based also. यहाँ से question I have seen in PYQs, especially I think 2021 if I am not wrong. So you have to remember all this, and the way it has been listed, this is listed in the order of their preference or availability in India or their use in India, right? So yeah, please remember the sequence also. The sequence should come like this only: Eisenia foetida, Ferritima elongata, Eudrilus eugeni, and Perionyx excavatus. This is in the order of preference as well. Fine. Benefits of vermicomposting, we all know. Vermicomposting mein, you can see it is a bit more than FIM. It contains micronutrients like this, growth regulators and beneficial microorganisms. Since it is coming out of the gut of the earthworm, it will definitely contain some beneficial microorganisms which will be helpful for the nutrient release and the plant growth. It increases water holding capacity of the soil. This is common for all organic manures, so this is nothing new. It increases aeration in soil, again common for all organic manure. Decreases salinity and increases availability of nutrients, same again. Reduces soil erosion, decreases cost of cultivation, reduction in incidence of disease and pest. This is also the same. So this one is specifically unique to vermicomposting only, this one, right? Now we have entered the domain of fertilizer. So manure khatam hua, organic khatam hua. Now we are coming to fertilizer. Fertilizer is definitely easier, you know. Fertilizer we all know. Fertilizer में ज़्यादा कुछ है नहीं, but यहाँ से question तो आता है. So we will have to read. There is all right. So fertilizer, we will see the definition of fertilizer. Yes, fertilizer का definition क्या है? Fertilizer का definition में you have to remember the keywords, because nowadays you know how the question comes. They will give you I will frame the question for you. Question: fertilizers are A, chemical, B, inorganic, C, organic, D, definite chemical composition, then E, low analytical value. So now you tell me which one of the following are true for fertilizer. This is the type of question NTA asks nowadays. I think all of you are very, very well aware with that now. Yes, all of you are very well aware with this pattern now. So this question I have framed it for you just here. Just read this definition and tell me which of the following about fertilizers is correct. Okay, Pawan is telling ABD. Very nice, Pawan. You're very attentive. It's so good to see. Yes, Raman, you are also exactly correct. So this is a very simple question, but it is just to give you a glimpse of how question might come and how you should be very thorough and how you should be very attentive while you are reading the definition. Exactly aisa hi question I have seen about minerals, you know. You have to remember the definition of the minerals. In that there are many keywords like inorganic, definite chemical composition, then और क्या था? Inorganic, definite chemical composition, and then those what is that called? Regular repetition in all the 3 dimensions, and all those were there. Yes. So similar question they have asked for mineral. They have given everything correct, and in case of inorganic they have made it organic, right? So ऐसा question अभी आ सकता है. Please be very thorough while you are reading this. So chemical substance, inorganic, definite chemical composition, and high analytical value. These are the keywords of fertilizer definition. हो गया. Classification of fertilizer. You can see all this. We will see. ये तो सर नाम से ही पता चल जाता है एक्चुअल में कि क्या बोलना चाह रहा है. Secondary fertilizer, it will supply secondary nutrient. Nitrogenous fertilizer will supply only nitrogen. P will supply only P. K will supply only K. Micronutrient fertilizer will supply micronutrient. Incomplete and complete fertilizer, when it will supply all 3 primary nutrients, it is complete. अगर नहीं है तो incomplete, like that. एक बार इसको देख लेना if you want. बाकी इसमें ज़्यादा कुछ है नहीं. Then classification of fertilizer. Classification of fertilizer. This I have taken from Dr. D.R. Vishwa's book. That is not needed for you right now, but once you come to MSc and you will give SRF, that book is necessary. So state fertilizer, fertilizer which supplies single primary nutrient. Again a keyword. These terms are very easy and you can just identify them by seeing the English word only. But the problem is these are very confusing. You get very confused. That is the problem. So बिल्कुल भी confused नहीं होना है. And you have to be thorough. Yes. State fertilizer मतलब only single primary nutrient. This is very important. N, P or K, not all. N or P or K. Okay. State nitrogenous fertilizer का जो fertilizer supplying only nitrogen to the plant. And यहाँ पे जो है you can see the examples, right? Then state phosphatic fertilizer, fertilizer supplying only phosphorus to plant, only phosphorus. You might ask me, sir यहाँ पे तो ammonium sulfate है, sulfur भी जा रहा है. Only one primary nutrient. You can see here, single primary nutrient. It can supply some secondary nutrient. Secondary nutrient के साथ हम लोगों का कोई problem नहीं है, but only one primary. Fine. Then state phosphatic, state potassic supply only potassium, that is potassium chloride, potassium sulfate. Fine. Then what is the definition of complex or compound? Complex or compound are both the same. Do not get confused. Kahin kahin mein you will find complex, in some other places you will find compound. Complex and compound are both the same. Yes. So complex or compound fertilizer ka kya definition hai? There also you have to be very thorough with the keywords. Fertilizer mineral containing 2 or more nutrients. This is the first keyword. 2 or more nutrients, of which at least 2 primary nutrients are in chemically combined form. Okay. These 3 are the keywords. Complex fertilizers are those containing 2 or more nutrients. Nutrients can be 2 or it can be more than 2. Lekin what is the next condition? At least 2 primary nutrients should be chemically combined. Should be in chemically combined form. These fertilizers are also called multi-nutrient fertilizers. So जो यहाँ पे square किया गया है, all these are very important. If you want to take a screenshot, you can take a screenshot. If you want to read this PDF after the class, you can read that. That will be very nice. So majorly, there are two types. NP complex fertilizer. इसका सबसे बेहतरीन best example है diammonium phosphate, which is probably the most used fertilizer in India, all across India nowadays. Most used phosphatic fertilizer. Yes, because SSP अभी और कोई use नहीं करता. SSP is low analysis and it is very hard to get. But DAP is probably the most widely used now. Then ammonium phosphate sulfate दो बार हो गया, sorry यार. Then nitrophosphate. Then another one is NPK complex fertilizer. In this NPK, all are there. How is this coming under this definition? See, this is coming 2 or more bhi hai. So it is coming in this definition only. But only 2 primary nutrients should be in chemically combined. At least 2. That too at least 2. It can be more than 2. But it should be at least 2 should be in chemically combined form. Okay. Iska to NPK 10-26-26-26. This is also a very popular fertilizer. If you do gardening and all in your home, you will see this is there. Then what is a mixed fertilizer? Mixed fertilizer ka definition kya hai? Mixed fertilizer ka definition hai fertilizer which are prepared by purely physical mixing of two or more straight fertilizers or 2 or more complex fertilizers. In some books you might find only straight fertilizers, but it is not so. It can be 2 or more straight fertilizers, it can be 2 or more complex fertilizers as well. But that is purely physical mixing, no chemical reaction is involved, no machine is involved, no factory is involved, nothing. Iska simple example hai, say you are applying urea, SSP and MOP to your crops, to your rice crops say. So for your rice, urea, SSP, MOP, if you want to apply it differently, alag se, if you want to apply it individually, you will take 3 times of labor. That is a waste. So what you will do? You will mix all the 3 fertilizers, you will calculate the calculation, the dosage and all, then you will mix all the 3 fertilizers and you will put it in the field. That is nothing but physical mixing, and that is called a mixed fertilizer. At that point of time, usually mixed fertilizers are done only just before application to the field, because you cannot mix it and store it. It might have some other chemical reaction the longer it stays, right? So it is usually done just before applying in the field. जो जो चाहिए उसको मिक्स कर लिया, उसको फील्ड में दे दिया. Yes, done. So इधर और 2 definition भी है. Incomplete fertilizer, the fertilizer which supply only 2 major nutrients. Here you can see NPK 12-32-0. यहाँ पे potassium नहीं है, no potassium. Then again complete fertilizer, the fertilizer which supply all 3 major nutrients, that is 15-15-15 NPK complex fertilizer. Fine. Then classification of fertilizer, we are going on. Fortified fertilizer, fertilizer with small fraction of micronutrient content. Boronated single superphosphate, zincated urea, zincated NPK. Then customized fertilizer, also called custom fertilizer, fertilizer developed for the need of a specific crop in a particular region. ये सब का बस you can remember one line. Question आएगा तो इसी में से आएगा, इससे बाहर नहीं आएगा. Then high analysis fertilizer, I think all of you know, fertilizer which contain fertilizer which contain more than 25% of major plant nutrients, that is a high analysis. Low analysis which contain less than 25%, that is a low analysis, right? We all know this. Moving on, nitrogenous fertilizers का ये classification है, you can read that. Nitrate fertilizers, nitrate, it contains nitrogen in the nitrate form. Then ammonium fertilizers, you can read ammonium sulfate, ammonium chloride, anhydrous ammonia, it contains nitrogen in the ammonium form. Then nitrate and ammonium fertilizers, it contains nitrogen in both ammonium and nitrate form. Then amide fertilizer, it contains in the NH2 form, and then cyanamide fertilizers, it contains in. Okay, is it okay? Everything is clear? I think यहाँ पे कुछ unclear होने का तो जगह ही नहीं है. Then synthesis of nitrogenous fertilizers. Yes, now here I want to say you something, जो you should listen. So synthesis of nitrogenous fertilizers यानी कि इसका जो processes है, these processes are not there in the syllabus of JRF. All right, these processes, specifically processes, are not there in the syllabus of JRF. If you analyze the previous year questions, you will rarely find there is no question regarding they are specifically asking cyanamide process का जो reaction है, from that reaction they are asking, it is not there, but it is there in SRF. Once you are applying for PhD entrance, it is definitely there. Okay, so now since it is not there in the syllabus of JRF, we will cover it only upar se. We will only cover the essential things, because even if it is not in the syllabus, you can find 1 or 2 questions from Haber's process if you thoroughly scan the previous year question. So Haber's process, they might consider it in nitrogen also. So that's why we will just have a overview of these processes. We will not go deep into the reaction, uska condition kya hai, but whatever is needed, whatever from which question can come, I have only synthesized those points. Okay, so we will have a quick overview of this. Is anyone there, or I think I am talking alone for so long? Please drop a thumbs up or anything, whatever. Please let me know that you are still there. Yes, Aishwarya, Pavan, very good. Okay, very nice, very nice. Thank you so much, everyone. So synthesis of nitrogenous fertilizer, the first process that is very important is the cyanamide process. Cyanamide process का जो reaction अगर तुमको पढ़ना है, if you want to read that, you can read that from old ISSS, बहुत अच्छे से दिया हुआ है. But I would suggest there is no need of that, so don't go into that. लेकिन cyanamide process में क्या याद रखना है? Those I have taken here. First point is not produced in India. You will see it is specifically written in the last part of calcium cyanamide process. It is not produced in India. Okay. Second point is developed in Germany, very important, by Professor Adolf Frank and Dr. Nicodem Caro. This is also very important. Who developed the cyanamide process might come. End product is calcium cyanamide. That is why the name comes, the cyanamide process. So end product is CaCN2, calcium cyanamide, easily converted into NH3. अल्टीमेटली हमारा नीड तो NH3 है, NH3 से NH4 होगा, NH4 से NO3 होगा, फिर प्लांट लेगा, CN2 को कन्वर्ट तो करना पड़ेगा ना. yes it बीच में 2-3 स्टेप है ना, यूज ऑफ यू हैव टू यूज कोक और कोल, दैट इज कार्बन, that carbon you should be calcined to very high temperature and all, those are not needed. Anyhow, this point is the most important point of the whole cyanamide process. You know, it is the first commercial process that was used worldwide to fix atmospheric nitrogen. This is the first process worldwide to fix atmospheric nitrogen. This is very important. This might come in true false, this might come in statement based question, anywhere. The product, that is this calcium cyanamide, it was known as nitrolim or lime nitrogen. Why? Because you can see यहाँ पे calcium है. Calcium के रहने के वजह से the product was called nitrolime or lime nitrogen. Fine. Cyanamide process से remembering this much is more than enough. You don't need to go further deep. You can be sure इतना सा अगर याद है तो you can make the questions in JRF. Okay. Not, I'm not talking about SRF aspirants. SRF may they ask it from deep, but JRF this much. Then second is electric arc process. I think all of you know this. This is also called Birkeland and Ide process. Yes. Christian Birkeland and Samuel Eide. It was developed in Norway. This is important. And the end product is nitric acid. This one, I think all of you know, this electric arc process is nothing but it is a mimicry of the lightning process that happens in the atmosphere. Lightning होता है, जो N का जो triple bond है वो टूट जाता है, the nitrogen becomes जो inert N2 form है वहां से reactive form में आ जाता है. That one single atom of nitrogen that will react with the rainwater and it will form nitric acid and that will come into the soil. That is how that chemical fixation of nitrogen occurs. We read biological nitrogen fixation. This is the chemical fixation, which is the like it जो energy needed है that is provided by the lightning. So this electric arc process is nothing but it is a mimicry of this lightning process where a huge voltage of electric is passed through nitrogen. That nitrogen is broken and then it reacts with water to form nitric acid. Yes. Yes, 3000 to 3500 degrees Celsius. That is why it requires huge amount of money because itna electricity produce karna, you can understand. It can only be done where there is cheap hydroelectricity available. For your information, Norway में जहाँ पे develop हुआ था, they had a hydroelectric company just beside their factory that was called Norsk Hydro, N-O-R-S-K, Norsk Hydro. So वहाँ से hydroelectric power लेके they used to do the electric arc process. Fine. Then third is Sarpek process. Sarpek process में क्या important है? End product is ammonia, important. It was given by Ottokar Sarpek, important. Nitrides of aluminium are used. Aluminium nitrides are used in this. This was also a very important or very known process during the World War times. Fine. Then Haber-Bosch process, we have to read this in detail. Whatever may be the scene, we have to read this in detail. Because Haber-Bosch process, it might come under the nitrogen also. And यहाँ से question तो मैंने देखा है. Still, I have tried to compile only the most necessary information for you. Okay, let's see. So Haber-Bosch process, the first important thing is it was given by Fritz Haber and Carl Bosch in 1913. Actually, it was given by Fritz Haber. The process was discovered by Fritz Haber in 1908, 1909, that period. Then Carl Bosch, he modified the process to make it more energy efficient and economically efficient in the year 1913. So it is usually taken as Haber and Bosch 1913. Okay, this process is taken like that. It is the basis of N fertilizer production today. Nowadays, everywhere where there is N fertilizer production, ammonia is produced in this process only. It is the basis of N fertilizer production today. End product is ammonia. You can see end product is ammonia. Power requirement is less compared to cyanamide and arc process. That is why it gained popularity. That is why everywhere economics is involved. जहाँ पे पैसा कम लग रहा है, that is economically feasible and that will definitely get popularity. Stoichiometric ratio of N2 is to H2 should be 1 is to 3. Here you can see. This is also a precursor. This is a very important point. This you should remember. You will see this reaction that I will explain later, don't worry. Then the pressure. Pressure should be 200 to 1000 kg per square centimeter. 200 to 1000 kg per square centimeter. Okay. Temperature required is 400 to 450 degrees Celsius. This is actually the major problem with nitrogen. The major problem with nitrogen, इतना fertilizer लेके लड़ाई, इतना कुछ क्यों हो रहा है? Because nitrogen का जो inert form है, this N2, this is a very hard molecule to break. उसका जो triple bond है, that triple bond has a energy of almost 10 electron volt, which is very high. उसको तोड़ पाना is very difficult. That is the reason why all this nitrogen लेके इतना सारा बात होता है. That is the only reason. Okay. Here you can see 400 to 450 degrees Celsius, 200 to 1000 kg per centimeter square pressure. This is also a huge amount. Anyhow, how much ever optimization you do for the process, you have to still provide a huge amount of energy. That is why this nitrogenase and this biological nitrogen fixation, it is like so astonishing a process, right? Like how the microbes are doing it, how they are doing it with the help of an enzyme that we humans cannot even do today. Still today we are fighting for nitrogen, we are fighting for nitrogen as fertilizer because you can see so much energy is required. Then the purity of gases should be as high as possible. this. Now please pay attention, whoever you are there, please pay attention to these last 3 points. All these 3 are purely exam oriented points. Catalyst is Fe oxide, iron oxides are used as catalyst, promoters are Mg, Al, K and Ca oxides. Magnesium, aluminium, potassium and calcium oxides are promoter. Please be attentive towards the difference. One is catalyst, one is promoter. Catalyst है iron, promoter है magnesium, aluminium, potassium and calcium. In original method, osmium, OS was used as the catalyst. Okay. In the original method, osmium OS was used as the catalyst, which was used by Haber himself. So Haber himself used osmium. He used to collect this osmium from electric bulbs, you know. He used to collect that osmium from electric bulbs. He used to work in a bulb company. From there, he collected and he applied that. So those are the stories of science. Okay, fine. So now please pay attention to the reaction. N2 plus 3H2. Please see the stoichiometric ratio. This you should remember. Then you can see the temperature. Then you can see the pressure. Yes, you can see the pressure. Fe oxides, these are the catalyst. Then aluminium, potassium, magnesium, calcium, these are the promoters. All right, promoters. Please remember this. Fine. Here you can see it is 2NH3 and it is a exothermic reaction. Exothermic reaction means it happens with release of heat. So this high pressure is required. High pressure is required. How you will remember that? Very easy. See here, 3 molecules of hydrogen and 1 molecule of nitrogen. Total 4 molecules are here on the left side of the reaction. On the right hand side of the reaction, only 2 molecules are there. So number of molecules or number of moles, it is decreasing as we are going on the right side. So decreasing का मतलब क्या है? The pressure should be more. If the pressure is more, the pressure is increasing, then only this reaction will proceed. Fine. Okay, I think we are clear. If you have any question, any doubt about this Haber's process, tell me. Please be very thorough with Haber's process. This is very important. यहाँ से question आने का बहुत ज़्यादा chances है. Okay. Another thing I would like to highlight here, very sadly I have to highlight this. Even after being clearly written as Fe oxides as the catalyst, Al, K, Mg, Ca oxides as the promoter, last year or last to last year in SRF, they have asked this question from Haber's process, same statement type question. There they have asked, Fe oxides, Al, K, Ca, Mg oxides are used as catalyst, and they have given that statement as the correct option. Okay. So NTA कब promoter और catalyst को equal मान लेगा, you never know. But luckily options were in such a way that you could answer that even without that statement. मतलब और कोई दूसरा answer था ही नहीं. Whatever was the answer in that statement was also included. So this is just for your information. But otherwise, from knowledge side, you should remember it is a catalyst Fe oxide. Promoters are Mg, Al, K and Ca oxides. Promoter और catalyst का क्या difference है? That we will study in the chemistry class. Okay. Now the nitrogenous fertilizers. In this also you have to remember the properties. जो properties से question आ सकता है, जो properties से question आ चुका है, I have only taken those properties. Fine, nothing to worry about. Nitrogenous fertilizers, urea, we all know, NH2, CO, NH2, 46% N. All of you know that. This is a question number 1. White in color, ये question कभी नहीं आएगा, don't worry. Hygroscopic equivalent acidity 80. I will just point out the questions. The first question, how much percent N, right? Then second question, equivalent acidity 80. Third question, CRH at 25 degree. CRH का full form please लिख दो यार comment में. बहुत देर से मैं अकेला बकबक कर रहा हूँ. What is the full form of CRH? Dear students, can you please write that? CRH का full form लिखो and can you make me understand what actually is CRH? Critical relative humidity, very nice. Can you make me understand what is a CRH? CRH 72 का मतलब क्या है? What is CRH 72? If CRH value is high, the hygroscopicity is less, or CRH value is less, the hygroscopicity is high. Do you know that? Can anyone tell me? What is a CRH? Yes, yes, Nikhila, Aishwarya. CRRI high, hygroscopicity high. Galat. It's wrong, Pavan, it's wrong. Inversely proportional. देखो, CRH का सीधा सीधा CRH वैल्यू हाई इज बैड. ऐसा होता है क्या? CRH value high is good. Here you can see CRH of ammonium sulfate is 81 and ammonium sulfate is the best structure wise fertilizer. CRH and CRH, here you can see this is a liquid anhydrous ammonia. और कुछ CRH है यहाँ पे. CRH 76. Here you can see. CRH जितना हाई है उतना अच्छा है. CRH जितना हाई है उतना अच्छा है. क्यों? Let's understand that. CRH is critical. CRH is critical relative humidity. We all know that. Relative humidity क्या है? I think ये सबको पता है. Relative humidity क्या है? We all know that. Amount of water vapor present divided by the amount of water vapor required to fully saturate that. We all know that. Relative humidity is expressed in percentage. So अभी का जो I stay now in Delhi. Delhi का relative humidity कम है. Delhi's relative humidity is less. But my hometown is in Bengal. It's in Kolkata. The relative humidity there is very high. So let us take a value. Relative humidity of Kolkata, say on a particular rainy day, the relative humidity of Kolkata is 90. Okay, let's take 90. Critical relative humidity means critical relative humidity of a certain fertilizer. Say fertilizer A. Fertilizer A, uska critical relative humidity of a certain fertilizer at 25 degrees Celsius. Temperature is very important in all the humidity calculations, as you know. At 25 degrees Celsius, the critical relative humidity of that fertilizer is 93. Okay. So 93 का मतलब है until and unless this relative humidity of the atmosphere will go to 93, it will not start absorbing water. Okay. So if the critical relative humidity is 100, that would be ideal because no atmosphere will be 100% humid. So ये दो value के बीच there should be equality. Until and unless this value reaches equal to this value, then only it will start absorbing water and it will become hygroscopic. Okay. So critical relative humidity value जितना high है, if it is 90, then that will be a very good fertilizer because वो कभी water absorb करेगा ही नहीं until and unless the atmosphere goes that high. Then say in case of urea it is 72. 72 का मतलब है when the atmospheric humidity reaches 72, it will start absorbing water. Okay, 72 से ऊपर जाएगा atmospheric humidity if it is 90, then it will absorb huge amount of water. But in case of ammonium sulfate, it is 81. So if the relative humidity of the atmosphere, say it is 70, in that case it will not absorb water. So the structure will be good. समझ में आया? Is it clear? Or do I need to tell again? Is it clear? Please tell me. Let me know. Is it clear, Aishwarya, Pavan, Shreyoshi? समझ में आया तो हाँ, very good. I can see thumbs up. बाकी लोग okay, so let's get back to the usual business of finding out questions and scoring marks. So question number 1, question number 2, question number 3, acidic in nature. This is also question number 4. ये भी आ सकता है ना? Isolated first from urine in 1773 by Rowell. Question number 5. First synthesized in lab by Wooler in 1828. This is also called Wooler synthesis. All right. Question number 6. First organic material. This is a very important statement. It is the first organic material synthesized from inorganic compound. If you see this Wooler synthesis reaction, उसमें पता चलेगा कि inorganic material से organic material was synthesized for the first time. So we get 7 questions from these points. ठीक है, याद रखना, ऐसे question wise भी याद रख सकते हो. It will be very easy. Then this biuret. In this biuret section, every word that is written, that is a question. I think all of you know, all of you have seen PYQ multiple times. Biuret से question आता ही आता है. Yes, Wooler made urea mistakenly. Correct, Pawan. From biuret, you will definitely get questions. If you have analyzed the PYQ, you must have got 2 to 3 biuret questions you must have encountered. All right. Whatever is written here, everything can be a question. This is the formula of biuret. You can see NH2, CONH, CONH2. It's almost like 2 molecules of urea. That is why it is called biuret. Greater than 2% biuret can damage the crops. Maximum permissible limit of biuret in urea is 1.5%. This is a very important question. Less than 0.5% for foliar spray. Very important question. Again, biuret sensitive crops like citrus and vegetables, less than 0.25% in foliar spray for citrus crops. These last 3 statements and everything written in this biuret portion, you can get questions and questions and questions. All right. Then coming to ammonium sulfate, इसका color होता है white, water soluble, non hygroscopic. You can see CRH value ज़्यादा है, so it is non hygroscopic. It is the best fertilizer when compared to structure and its physical characteristics. Equivalent acidity is 107. This is a problem. Acidity थोड़ा ज़्यादा है, acidic का problem है. 20.6% N. Again another question. 24% sulfur. Another question. Acidic problem. This is the problem. Acidic fertilizer. Okay. All right. This is the best fertilizer for application in which crop? Can anyone tell? That is also written in ISSS. That I have not taken. Ammonium sulfate is best for application in which crop? Which we regularly used to apply actually. Now it has been less due to the advent of urea. Legumes and crucifer. Okay, you are telling from sulfur point of view. That is correct. But the answer that I was wanting was, all right. Then coming to ammonium nitrate. It is also same, white, water soluble, highly hygroscopic. So problem of storing, transport and storage, equivalent acidity 60, CRH is 59. You can see the CRH value is very low. Yes, very nice. Divya and Nikhila, very nice. Paddy. So again, I think by now you understand the concept of CRH. So CRH itna kam hai, matlab whenever the atmospheric humidity reaches 59%, it will start absorbing water. So highly hygroscopic, problem of storing is huge, transport and storage problem is huge, equivalent acidity is 60. CRH is 59, okay. 33 to 34% in acidic fertilizer, but not that much acidic as compared to urea and ammonium sulfate. The another major problem is explosive. This is also a question because ammonium nitrate, I think few days back you might have seen in news one fertilizer factory that blasted in somewhere, I don't remember the country, maybe in Oman or somewhere near to Turkey maybe. So it is highly explosive and storing, that's why it is a huge problem. So you cannot get ammonium nitrate that much in the markets because if you store it mistakenly, it will explode, okay. Calcium ammonium nitrate can, very important part. यहाँ से question मान लो आ ही गया, question has already come. हाँ, yes. Delhi blast अभी कुछ दिन पहले हुआ था ना, in front of Red Fort. Yes. So gray or light brown, partly soluble. less hygroscopic than nitrate but more hygroscopic than sulfate. Okay, less hygroscopic than nitrate but more hygroscopic than sulfate. 25% NEN, 25-26 likha hai, but majorly we take it as 25. This is the most important part. It is absolutely neutral fertilizer. That's why the equivalent acidity is 0. Now you have to tell me what is the name of CAN in Hindi. This is also written in all the books and they ask the question. No, Bahauddin sir, I cannot explain. I cannot explain to the highly intelligent people. Kisan Khad, exactly. This is also a very important question that might come in your JRF. Okay, so please be aware. Now coming to ammonium sulfate nitrate, you can read. This is not that much important, so I will skip it simply. हाँ, किसान खाद का अगर हम translate करेंगे तो its literal translation translates to farmer's fertilizer, right? Kisan khad. Kisan is farmer, khad is fertilizer. It is neutral, equivalent acidity is 0. These things you should remember. Then coming to ammonium chloride, equivalent acidity is 127, CRH is 76, least preferred fertilizer, you know, because of this huge amount of acidity. Then anhydrous ammonia, the equivalent acidity is 148 and इसका CRH value नहीं लिख सकते because it is already a liquid. A liquid cannot absorb more water. All right. So here I have done this for you. The amount of percentage it is there, you can see. So in this, can you please do a thing for me? List on ascending order of acidity. Can you list? अमोनियम नाइट्रेट कोई नहीं भाई, किडिंग ऐसे अभी concentrate करो, concentration is more important. All right. Ascending order of acidity. I will give you the names of the fertilizers. Can you please write in ascending order? Ascending order of acidity. You just have to remember the equivalent acidity value and you can write it very easily. There is no problem. Pawan, please do not make jokes like that. This should be an open platform for everyone and please do not make other people feel bad with your jokes. Anyhow, can ammonium nitrate, urea, ammonium sulfate, ammonium chloride. Okay, and this also you can add in the last, right? This also you can add in the last. So yes, Pawan, you are absolutely correct. I will can ammonium nitrate, urea, ammonium sulfate, ammonium chloride, anhydrous ammonia. Perfect. So all of you are on the perfect right track. I'm very happy to see that. Can is having the least, right? Then ammonium nitrate, of course, then urea, then sulfate, then chloride, and then anhydrous ammonia. So I can see only 2 answers. That is a bit heartbreaking. I expected from other people also. Nikhila and who was there? Divya. Fine. You can still write, no problem. So coming to the slow release nitrogenous fertilizers, it releases nitrogen slowly in available form to develop materials which control the release of N in available forms very slowly. Why is this needed? because थोड़ी देर पहले I was telling that the problem with nitrogen is huge. We cannot enhance the efficiency of the nitrogen. Nutrient use efficiency of nitrogen is very less. That is the major problem. Okay, we are applying 100 kg nitrogen, उसमें से 70 kg is getting lost to the environment through leaching, through volatilization, through emission of nitrous oxide, जिसके वजह से environment pollution भी हो रहा है, and the crop is also not getting the nitrogen, and also the cost of application of fertilizer is also increasing. So there are many problems. So just for solving that problem, पिछले 20 to 30 years research has been going on. What to use? I think all of you know nitrification inhibitors, urease inhibitors, sparingly soluble fertilizers, coated fertilizers, whatever and all. Our only goal is to slow down the release of nitrogen, since nitrogen is highly mobile and highly prone to losses. That's why our only goal is to slow down that nitrogen release so that it is in sequence with the crop growth, and in sequence with the crop growth it will release the nitrogen which will be taken up by the plant and the loss will be less. लेकिन इतना कुछ करने के बाद भी 40% से ज्यादा we are still not getting. That's the sad part. Okay. So what are the advantages? Minimizes N loss, increases the efficiency, avoiding frequent application of N fertilizer. Yes, because we have to go for split application of N fertilizers. We all know, because basal application में अगर सारा का सारा dose डाल दिया तो उसमें से सब कुछ चला जाएगा. We will have nothing for the plants. That's why we go for split application. We try to do it according to the growth stages of the crop, but still the problem is same. So here you can see avoid frequent application of N fertilizer. If it is a slow release fertilizer, it will release it slowly, and one application in the first time or application one time only, single time only, it will go on releasing. Fine. Avoids luxury consumption of N. Then classification, conventional soluble N fertilizer coated with some other material. Okay. Just I was telling, coated fertilizer. Then N substances of low water solubility. The examples are very important. Please see the examples. Sulfur coated urea. Here it is urea formaldehyde, also called urea form. Then crotonylidin urea. All this इसका जो nitrogen percentage है, that also I would suggest you please see once from treatise. Treatise में है. These questions regularly come in SRF, but in JRF these questions don't come. So I have avoided these portions just to make you feel better. The nitrification inhibitors, इसका examples तो very important है. Nitropyrin, NSAR, VM. Can you write me some natural nitrification inhibitors? 1 or 2, whatever you want. Pawan, Shreyoshi, Aishwarya, Nikhila, anyone? I mean all of you. Can you please write some natural nitrification inhibitors? These two are chemicals. इसके साथ एक और बहुत important chemical भी है, that is called DCD, dicyan diamide. Yes, dicyan diamide, DCD is also very important. But all these three are chemicals. All these three are chemical compounds, right? Yes, all of you are exactly correct. Neem and Karanj. Neem and Karanj are two of the most brilliant, highest efficiency natural nitrification inhibitors. Here you can see sparingly soluble minerals. This also you can apply somewhere, but this will have very low release and probably your crop will not be able to take up the nitrogen as per the requirement. Okay. So we have come to phosphatic fertilizer. I think Aishwarya, now your doubt will be better clarified. Phosphatic fertilizers on the basis of forms in which orthophosphoric acid is combined with calcium, water soluble or monocalcium phosphate. Okay. इसका example है SSP, DSP, TSP, single superphosphate, double superphosphate and triple superphosphate. Then citric acid soluble dicalcium phosphate, that is basic slag dicalcium phosphate. Then insoluble in both water and citric acid, that is citric acid insoluble phosphate. उसका example है रॉक फॉस्फेट, रॉ बोन मिल, स्टीम्ड बोन मिल. इसके बारे में हमने बहुत ज्यादा चर्चा किया. Out of 25%, only 16% is citrate soluble, and in this out of 20%, only 8% is citrate soluble. Baki, the majority is still insoluble in both citrate and water, right? Then phosphatic fertilizer contains the nutrient phosphorus in adsorbable form, that is the phosphate ions. इसका सबसे जो important example है, that is of course single superphosphate. ये जो सुपर है, ये सुपर का कोई ज्यादा मीनिंग मत निकालना. This was called super because it was very easy to make and it was in very high use. That's why they named it as superphosphate. 16% P2O5. Please be very careful. 16% P2O5, not P. अगर P पूछा तो divided by 2.29. 12% S, 19.5% calcium. It is prepared by rock phosphate plus H2SO4. Very important. Rock phosphate plus sulfuric acid. Very, very important. Hygroscopic, water soluble, yes. Gray ash-like powder. I think all of you have seen it. Even if you have not seen it, it is gray color. Powder, full it is like powder. Good keeping or storage quality. P fertilizer hardly moves in the soil and hence they are placed in the root zone. That too, they are only applied as a basal application and इसका split application का कोई जरूरत नहीं है because basal application is enough to supply for the whole cropping cycle, even for the next cropping cycle. Okay, then triple superphosphate is the next. Triple superphosphate contains 46% P2O5. Okay, this is prepared by rock phosphate plus H3PO4 phosphoric acid. ये दोनों तो एक क्वेश्चन है भाई. This is question number 1, this is question number 2. These two might come in this year's JRF also. Please remember. Yes, it is first fertilizer produced in India. It was the first fertilizer produced in world as well. Yes, Vipin. Suitable for all crops and all soils. In acid soils, it should be used in conjunction with organic manure. Rock phosphate has 25 to 40% P2O5. Citrate soluble is only 5 to 17%. Applied in hot, humid climate, low P, broken farm, and only in acidic soil. Because if the soil is not acidic, it will not be able to release the phosphorus from the rock phosphate. Yes, because it is a mineral and the phosphate is in highly structurally adsorbed form and it is very hard to release that. Okay. Then complex fertilizers, we have already read ammonium phosphate, H3PO4 plus NH3, monoammonium phosphate, that is 1152.0. This is very important. Monoammonium phosphate matlab one ammonium, one phosphate, right? Here, जितना भी यहाँ पे लिखा है, only in these last 3 lines, all 3 are very important. Monocalcium phosphate, this rating 1152.0 important. H3PO4 plus NH3, this is important. Acidic pH 3.5, this is also important. Moving on, diammonium phosphate, definitely you can see 1:2. So diammonium, 1 phosphate and 2 ammonium. Diammonium phosphate 1846.0, very important. Slightly alkaline pH 7.9. Ammonium phosphate sulfate, it is a mixture of monoammonium phosphate and ammonium sulfate that forms ammonium phosphate sulfate. Then urea ammonium phosphate nitrate. All these are the complex fertilizers. Okay. Then phosphatic fertilizers का एक बहुत important property है this band placement. Band placement is a method where fertilizers are applied in narrow bands. Fertilizers are applied in narrow bands or lines in the soil, usually close to the seed or plant roots, instead of spreading them uniformly over the entire field. Because if you spread phosphatic fertilizers over the entire field, entire phosphorus will get absorbed. And since we have read in the last class only, phosphorus H2PO4 minus comes in. According to Bray's concept of nutrient mobility, it is a mobile nutrient or immobile nutrient in soil. Can you tell me? It is an anion. It is an anion. So phosphate is mobile or immobile in soil? Anion है तो क्या होना चाहिए? Phosphate, according to Bray's concept of nutrient mobility, is it mobile in soil or immobile in soil? Pawan is writing immobile. What about the other guys? Yes, immobile. Bah, very nice to see that. Even with my deliberate try of confusing you guys are not getting confused. That is a very good sign and very nice. So this is immobile, highly immobile because of ligand exchange and specific adsorption and all those processes gets absorbed. This is a highly immobile nutrient. As a result, इसको अगर root zone के पास नहीं रखा गया, if it is not in the root surface sorption zone, तो इसका absorption by plant nahi hoga. It will not get absorbed. That is why band placement is needed, where it is applied in narrow bands in the soil, usually close to the seed or the plant roots, so that with the expectation that it will get absorbed, since it will be in the root surface sorption zone and not in the root system sorption zone. अगर हम broadcasting करेंगे, spreading uniformly करेंगे, तो it will get absorbed and It will be in the root system short-sun zone, but it will not get absorbed by the plant. Okay. So advantages of band placement: higher efficiency of phosphorus use, better root development, reduced fixation loss. See, everything is written here. This is prepared so nice. Requires less fertilizer quantity compared to broadcasting, particularly useful in low P soils. Again, I have to say thank you to Rohit. This PPT was made by Rohit. It is very nice. Then finally, we have come to potassic fertilizers, chemical substances containing potassium in adsorbable form. In absorbable form. Okay. So इसका सिर्फ दो ही आता है. All of you know KCl and K2SO4. KCl is the muriate of potash, MOP. White or red. You have seen the color probably. Pinkish color का आता है. Crystal containing 60% K2O and chlorine 47%. Completely soluble in water. Very important. Therefore readily available to the crops. Important. Not lost from the soil as it is adsorbed on the colloidal surface. Again the same Braze concept of nutrient mobility. Then it can be applied at sowing or before or after sowing. Chlorine content is objectionable to some crops like tobacco, potato, etc. All of you know this because tobacco, uska burning quality poor ho jata hai, and in case of potato, uska starch quality ghat jata hai. It becomes very sugary. So for tobacco and potato, we should use SOP, sulfate of potash. But be very honest with you, कोई भी farmer ये use नहीं करता है. I know many potato farmers who just apply KCl only because SOP is highly expensive and it is very hard to get also. So remember, how much ever theory you read, whatever you do, practically always there is a constant, and that is the economic constant. सारा पैसे का खेल है ना. Potassium sulfate, white salt containing 48% K2O, very important. 48, sometimes it is written as 50 also. So please remember the range. 48, 50, kuch bhi ho, both are there. 50% K2O. Again, I am telling you, all these are the K2O. All these I am talking percentage of K2O. If they are asking K, you have to divide it by 1.2, है ना? Soluble in water, readily available to the crop, does not produce any acidity or alkalinity in the soil. Preferred for fertilization of crops like tobacco, potato, etc., where quality of prime importance. Costly. It is costly. See, it is costly because it is made by treating potassium chloride with magnesium sulfate. So, chloride sensitivity. Yes, very nice, Aishwarya. So, chalo, ho gaya. Now, these are the some important terms which we should definitely remember. ये सारे टर्म्स याद होना ही चाहिए, है ना? ये सारे टर्म्स याद होना चाहिए. These are very easy terms, nothing is there. So what is the fertilizer grade? Fertilizer grade क्या है? It is the guaranteed minimum percentage. It is the guaranteed minimum percentage of the nitrogen, phosphorus and potash contained in fertilizer material, also known as the NPK rating. Guaranteed minimum percentage का मतलब क्या है? If the rating is needed as 28280, it indicates that 100 kg of fertilizer material will definitely contain 28 kg of nitrogen and 28 kg of P. Okay. So guaranteed, it is the guaranteed minimum percentage. So fertilizer rating में अगर बोल रहा है 60, so it is guaranteed that 60% K2O is there. That means 100 kg fertilizer डालने से 60 kg K2O तो मिलने ही वाला है. Okay. So that is called fertilizer grade. It is the guaranteed minimum percentage. Okay. Please remember the keyword, guaranteed minimum percentage. Then the number representing the grade are separated by hyphen, always stated in the sequence of N, P and K. Yes, of course, NPK, har jagah mein NPK. So it is always stated in the sequence of NPK. Label on the fertilizer bag with the grade 28280 indicates that 100 kg of fertilizer material contains 28 kg N, 28 kg P and no potash. 28 kg P2O5. Okay, very nice. Then fertilizer ratio and fertilizer grade are very similar, but please don't get confused. Both are different things. Yes, fertilizer grade and fertilizer ratio. Please do not get confused. Fertilizer ratio is nothing but it is the ratio of percentage. It is the ratio of percentage of N, P2O5 and K2O in the fertilizer mixture. All right, is it clear? Fertilizer ratio, it is the ratio of percentage of N, P2O5 and K2O. Fertilizer grade 1266. 1266 का मतलब है This is the guaranteed minimum percentage. 100 kg fertilizer डालेंगे तो 12 kg N, 6 kg P2O5, 6 kg K2O. But इसका ratio, fertilizer ratio अगर देखेंगे तो you can see all this is divisible by 2. So 12 by 2, 6 by, sorry, all this is divisible by 6 only. 12 by 6, 6 by 6, 6 by 6. So the ratio is 2:1:1. So it indicates that in this fertilizer, nitrogen, phosphorus and potassium is available in the ratio of 2:1:1. So how much ever you supply 50 kg, 100 kg, 200 kg, the ratio of the nutrients will be 2:1:1. Okay. Now what is conditioner? Conditioner is a low grade. These are important keywords. Again, I am coming in the keyword approach. Conditioner are the low grade organic materials like peat soil, paddy husk, groundnut shell, etc., which are added to fertilizer mixture during their preparation for reducing hygroscopicity. This is very important, reducing hygroscopicity and to improve their physical condition. Okay. Filler. Then what is a filler? What is the difference between filler and conditioner? Please pay attention. Filler is a wet makeup material. It is a wet makeup material. For making this ratio, for making this grade, you need some makeup material because exact chemical combination of all the material is not at all possible in the real world. तो उसके लिए हमें क्या चाहिए? कुछ filler material चाहिए. हम बोलते हैं ना, when we are playing music, in between two songs, they tell कि कुछ filler बजा दो. Filler का मतलब है उसका कोई function नहीं है, उसका कोई role नहीं है, but it is just making up the time. Here it is just making up the weight. So it is a wet makeup material like sand, soil, coal powder, ash, etc. added to the fertilizer ingredient so as to produce a mixture of the desired grade. Desired fertilizer grade पाने के लिए we have to use some fillers. So these fillers and these conditioners, especially these fillers, they should have some specific properties. वो specific property क्या होगा? Like it should be non-reactive, it should be cheap, it should be easily available. All those properties, those are common sense properties, nothing to read there. That's why I have not added them here. So finally, we have come to green manuring and biofertilizer. So green manuring, we were talking in the initial slides only. Green manuring is the practice of enriching soil by growing of a crop and plowing in situ or turning it into the soil as undecomposed green plant material. इसका मतलब क्या है? The soil or the land patch in which we need to grow our plants, in that we will grow these green manuring crops. Then just before flowering or during the flowering stage, we will plow all the crops. Like the crops are standing in the field, right? The crops are standing in the field. We will directly put a plow and incorporate that crops directly into the soil. It should be done before flowering or during the flowering stage specifically because uske baad the CN ratio gets high and it will take huge time to get decomposed and the nutrient will not be released that fast. So यहाँ पे एक और चीज याद रखना, flowering. The turning in should be during the flowering phase or before flowering phase, just before flowering phase. उससे बहुत ज्यादा आगे अगर कर दिए, like if you do it too early, then also you will not get the desired amount of nutrient because तब तो वो उतना बड़ा हुआ ही नहीं है. It has not accumulated that much nutrient, so it defeats the purpose. And if you are doing it too much late, then also it is defeating the purpose because तब वो release नहीं कर पा रहा है. Okay. Then the purpose is to improve physical, chemical and biological properties as well as the fertility of the soil. Green manure crop supplies organic matter as well as additional nutrients, particularly nitrogen if it is a legume crop. So most of the green manure crops are legume crops so that it can add extra nitrogen, which has the ability to fix nitrogen from the atmosphere with the help of its root nodule bacteria. जिसका नाम है rhizobium, all of you know. The practices of green manuring are classified as in situ and ex situ. In situ and ex situ में क्या difference है? We'll see here. In situ practice, green manure crops are grown and buried in the same field. We all know what is the meaning of in situ. In situ means in that place only. So crops are grown and buried in the same field, either as pure crop or as an intercrop with the main crop. Pure crop बना के कर सकते हो if you have some gap between the crops. If you have some gap available, in that you can take green manuring crop as pure crop. And if that gap is not available, you can take green manure as intercrop also, so that you can plow the intercrops only and incorporate them into the soil. Okay. The commonly used green manuring crops are sun hemp, very important, Crotalaria jantia, then dhaincha, Sesbania aculeata. From this many questions come, you all know. Green manuring crops ka example se. Then cluster bean, Melilotus, horsegram, cowpea, lucerne. You can read the examples. All the examples are very important. Then in case of ex situ green manuring, which is also called green leaf manuring, in this practice, plants that are grown elsewhere are brought in for incorporation into the soil as green leaves or tender green twigs. So these are usually big plants जिसका पत्ते लेके collect करके लाया जाता है and that is incorporated into the soil. So which are the most common examples? Leuciana leucophila, subabul, Glycidia, then Sesbania speciosa, and then Pongamia pinnata, Karanj. We were talking, Karanj is nitrification inhibitor, right? So all this, you can see, all these are big trees. Yes, are bade ped hai. So these big trees, definitely you cannot put it into the field and then plow it, right? So this tree is grown somewhere else. We collect the leaves and the green twigs and the young twigs of that and we incorporate it into the soil, okay? Green leaf manuring, this is extremely important, you know. That's why specifically I have added another slide. This is extremely important. What should be the characteristics of the green manuring crops? You can expect, definitely expect question from this. All these 7 statements, from this they can ask any 4, any 2, any 3 statement and they will tell which is correct, which is not correct, which of the following are correct, like that, okay. Legume with good nodulation, good nitrogen fixing capacity, number 1. It should be fast in growing even under water and nutrient stress conditions. This is the major criteria, because if it is not growing fast, we do not have enough time. We do not have enough time between 2 crop cycle to incorporate it. It should be very fast. Then low water requirement for its own growth, because again it is just used for improving the soil. So we cannot spend our money like a original crop, like जितना हम rice या जितना हम wheat के लिए spend करेंगे, उतना we cannot spend for the green manuring crop. That is as simple as that. So it should have low water requirement. Then it should be deep rooted system to absorb moisture and nutrients from deeper layers. So in short, it is trying to tell the plant should be hardy. कितना भी stress condition हो, it should be able to take up the nutrients from anywhere. The tender leaf permitting rapid decomposition. Leafy habit or tender leaf. This I was telling. It should be leafy habit or tender leaf so that it will permit easy decomposition and easy release of nutrients. Then short duration. ये तो already यहाँ पे बोल दिया गया. It is a short duration, fast in growing, short duration. Then rapid decomposability, non-fibrous and succulent. जितना succulent होगा उतना moisture होगा, उतना easily decomposable होगा. Non-fibrous होगा उतना easily decomposable होगा. Then it will release the nutrients, right? I think it is clear. And also I told, please remember that it should be done before flowering or at the time of flowering, not after that, because it will become fibrous. Then these are the advantages of green manuring. These are not that important. You can read advantages of green manuring. We all know what are the advantages of green manuring. Increases the organic matter, definitely. Returning the different plant nutrients to the soil, improves the soil structure, aeration status, all that are the same for all organic manure. Reduces the soil loss caused by runoff erosion. Yes, because it is a crop, it has root, it will hold the soil. Due to green manuring, the nutrient regime can be improved and restored, which otherwise would have lost by leaching. Green manure crops have some residual effect in relation. So these are all simple things, nothing question worthy. Yes. So we have come to biofertilizer. What is a biofertilizer? Here also sometimes confusion happens. So for all of you, I know who you are preparing very nicely. For them, it might not be a confusion, but who of you who are preparing, say, just from few days back, so biofertilizer, the term might have some confusion for you. So please do not be confused. Biofertilizer is a preparation containing primarily the active strain of right type of biological agents or microorganism in sufficient number. Biofertilizer is not a fertilizer. Biofertilizer is nothing but living organisms. ये question आता है as a true false, as a statement type question. Biofertilizer is actually living microorganisms. Biofertilizer के साथ fertilizer का कुछ लेना देना नहीं है. Okay? These are microorganisms. Used for enhancing crop production by increasing the biological activity of the microbial inoculant in the root environment. Biofertilizer may be carrier based or liquid form. This is also important. Biofertilizer may be carrier based or liquid form. It converts non-usable form to usable forms through biological processes. It converts non-usable nutrient forms, like say the nitrogen in a organic manure that is fixed as a protein, that is fixed as a complex polysaccharide, that is fixed as a huge macromolecule. उसको उस नॉन यूजेबल फॉर्म को it will change to usable form through biological process. For that we use biofertilizer, and there is nothing else. There is no fertilizer, there is no manure, that is just living microorganisms. Okay. Use of biofertilizer is the inoculation of desired microorganism in soil either directly or with the seed or planting material called as soil or seed inoculant. I think you might have seen before planting we use root dipping technique. Before planting, seeds को biofertilizer में डुबो के ऐसा किया जाता है and then we plant. Okay. When inoculants is a bacterial strain, preparation is termed as bacterial fertilizer or bacterial biofertilizer, and the use is called bacterization. Similarly, in case of algal strain, the preparation is known as algal fertilizer. These are common sense things, nothing to do. Advantages also, all are common sense advantages. I don't think anything needs to be explained here. Renewable source, help to improve nutrient status, long-term soil fertility and sustainability, increase in yield, chemical fertilizer can be minimized, no adverse effect on plant growth, eco-friendly, no pollution, energy requirement for production is less, production cost is less than chemical fertilizer. This is important. Again, cost is involved. Yes. Cheaper and cost effective. Some of the biofertilizers like BG and Azolla can be multiplied by the farmers in their own field. So it can be grown in situ and directly applied into the field. खरीदने का भी जरूरत नहीं है. Then rhizobia. Rhizobia तो हर जगह चला आता है. Rhizobia we are studying from last 2 classes. Rhizobia is a generic name for a certain gram negative. Very important. याद रखना इसको भाई. Gram negative, very important. Group of alpha proteobacteria and beta proteobacteria that can form nodules on the root, we all know, or in some cases on the stems, Sesbania rostrata. This is also very important. Nodulation on the stem, that is Sesbania rostrata, and it can fix nitrogen symbiotically. Among these, the genus Rhizobium is the largest genus of Rhizobia, having 112 species. Okay, that is for information. यहाँ से question नहीं आएगा. Cross inoculation. Cross inoculation is very, very important. You can expect match the following from cross inoculation अभी से, है ना? Cross inoculation group refers the group of leguminous plant that will develop effective nodule when inoculated with the Rhizobia from the nodules from any member of that legume group only. So cross inoculation means there is specificity. Not every Rhizobia will infest all the legumes, all the roots of the legumes. So there is specific strains of Rhizobia which can infest specific species of crop. That is called crop inoculation, cross inoculation. So can anyone tell me which are the compounds that results in this specificity? This is written in the soil biology chapter. I think you have read by now. Can anyone tell me which are the compounds that results in this specificity of the Rhizobium with the plants? Which are the class of those compounds? Flavonoids and isoflavonoids. Very good, Pawan. Yes, Vipin, these are non-spore and gram negative. Very nice. Flavonoids and isoflavonoids. Yes, Nikhila, you are also correct. Node factors, this is also important, but I think this is a bit high level, not for JRF, but still you can see. It is produced by Rhizobia in essential signaling component for symbiosis development. Node factors from different Rhizobia recognize different, see here it is written, different flavonoids secreted by different legumes, and this host specificity recognition. This is the reason. Okay. So again, this table I think I am covering this table for the 3rd time now. Every day this table comes, and this table comes this many times because it is that important. Again, I am telling you, ये जो table जिस form में है, in the exact same form you can expect match the following that might have already come in this year's JRF, है ना? So Meliloti, again I will say the same thing. Just by reading the species name, you can identify. Meliloti is related to Melilotus. Trifoli is related to Trifolium alexandrium, that is clovers. Okay. Then Leguminosarum is related to the legumes. Then Phaseoli is related to Phaseolas, that is the beans. Then Lupini is related to Lupinas, that is lupines. And then Japonicum is related to soybean. Okay. This is again, I am covering this for the 3rd time. So moving on, this is the formation of root nodule. This most probably will not come. This is not in JRF level, but still I will say, you can read this a point. This is nothing, it's like chemical signaling only. Flavonoids and isoflavonoids will be released, the bacteria will come, the bacteria will start infesting the root hairs, then the root hairs will be curled like this, that is called shepherd's crook. Then with this curling only, this rhizobium will digest the cell wall and enter into this. Then they will develop the infection thread. That infection thread will go into the cortex. In that cortex, de-differentiation of cells will happen. Then it will form the nodules. So do not think that these nodules are the bacterial cells or these nodules are the bacteria. Nodules are nothing but extension of plants only. Plants ne hi wo nodule banaya, uske andar that bacteria is living. All right. So enlarged root cells only form a nodule. Okay. And yes, Pawan has written active site of nitrogen fixation is bacteroid and it is separated by a peribacteroid membrane. Yes, all right. Peribacteroid membrane. We all know that. Fine, very good. यहाँ से इतना ही जरूरत है, बाकी अभी रिविजन में और इससे डीप नहीं जाएंगे. In the next year, if you guys are coming to IRI and you are taking my NET or SRF course, in that we will have a huge discussion, maybe one whole class on this. And for the who you are watching for next year JRF, in the master course also we can discuss. So these are the stages in root nodule formation. It will be recognition, then excretion of nod factors, then invasion of root hairs, then traveling to main root via the infection thread. This infection thread term is very important. Infection thread term is very important. Red color in active nodule is due to leg hemoglobin, which helps to prevent oxygen interaction with nitrogenous enzyme because nitrogen is oxygen sensitive. Very nice. But the color is not red, it is pinkish, you know. If you have taken up a legume and see the root nodules, it will be pinkish. Or books mein bhi main pink hi dekha. It is not exactly red. Okay, fine, very good. Formation of deformed bacterial cells called bacteroids within the plant seeds and development of the nitrogen fixing state. Continued plant and bacterial division forms of mature root nodule. This bacteroid is the seat of N fixation. This is also another question I am telling. Here goes your 4 marks. This question I have seen many times, many times. This question I have seen many times in previous year papers. The seat of N2 fixation, the seat of nitrogen fixation, everything, सबका answer होगा bacteroid, bacteroid. All right. Please remember this. Flavonoid, isoflavonoid, bacteroid. ये 3 term तो याद होना ही होना चाहिए. Fine. Then process of N2 fixation. N2 fixation is mediated by the enzyme called nitrogenase. We all know that, which mediates the reduction of N2 to ammonia. First, this enzyme was extracted from Clostridium. This is for your information. Nitrogenase is a functional enzyme which reduces N2 to ammonia and depends on energy source from ATP, definitely. Nitrogenase has 2 components. This is important. Nitrogenase has 2 components, one containing MOFE protein cluster and the another called as MOFE protein and the other only Fe protein. And remember, in both these clusters, sulfur is common. So it will be MOFES cluster and another will be only FES cluster. So sulfur is common in both. That is also a function of sulfur. Why sulfur is needed so much in case of legumes and oilseeds? Reduction of N2 to NH3. Again, this one comes again and again. It takes place with the help of the enzyme nitrogenase. So I was telling you, right, we are spending so much money, 400 degree, 450 degree, 1000 kg per centimeter square pressure just to fix nitrogen, and prokaryotes are doing this just with the help of one enzyme. Nothing else is required. Simply, it is still an energy extensive process, I agree, but it is happening in the soil naturally. So how helpless we are in front of nature, right? We cannot do anything. If any one of you can find a way to install this nitrogenase in some other animals or humans or other plants, then nitrogen ka samasya will dur ho jayega bhai. Let's see. Reduction takes place on the surface of the enzyme. So this is the step. Yesterday I was also talking about hydrazine. I remember in the nitrogen cycle, this hydrazine is toxic to many organisms. Then the overall biochemical reaction, this reaction is coming again and again and again. I will tell you again. This from this single reaction you can get at least 3 questions. So please remember all this: art electron, solar ATP, all this should be on the tip of your fingers, right? Then nitrogenase, then 2 moles of ammonia and N2. Fine. Then azotobacter, azospirillum, frankia, all these are biofertilizer only. We are still in the biofertilizer domain only. So azotobacter fixes atmospheric nitrogen non-symbiotically. Non-symbiotic का सबसे बढ़िया example है azotobacter. I think all of you know that, yes. Then the extent of fixation is directly dependent upon the amount of carbohydrate and utilized by them. It is an aerobic, chemoheterotrophic and free living bacteria. All these three are extremely important. All these three keywords are very, very important. If you want to take a screenshot, but you have to remember these three. You have to remember these three words while you are reading about azospirillum. Okay? All these three are question worthy words. They work better in the root region of crop non-symbiotically when adequate organic matter is present. That is because the roots will release some exudate, the root will release some organic matter which will provide the food for azotobacter. बाकी उसका कुछ symbiosis नहीं है. Sir, I read somewhere that azotobacter is associative as well. No. Associative तो azospirillum है ना. Azospirillum is associative. Azotobacter is purely non-symbiotic. And azotobacter के बारे में another information I will give you. Azotobacter has the highest metabolic rate of everything in the plant and animal kingdom. अब ये क्यों है इसका ये reason है. Azotobacter does not have access to leg hemoglobin, right? Leg hemoglobin is only in legumes. तो राइजोबियम तो लेग हीमोग्लोबिन के सहारे ऑक्सीजन से बच रहा है. राइजोबियम is getting rid of oxygen with the help of leg hemoglobin. He is very happy. But azotobacter का क्या होगा? He is alone. He is free living in the soil. Who will save azotobacter from oxygen? For that reason, azotobacter has developed many adaptations, like what happens in nature, evolution and adaptation. So azotobacter has got the highest metabolic rate among all of the plant and animal kingdom because that highest metabolic rate वो जितना भी oxygen आता है उसको respiration से use करके it uses it so fast that oxygen is unable to reach the nitrogenous. Okay, ये एक हुआ. Second, Pawan is writing slime layer. It develops a slime layer across its cell wall so that oxygen cannot penetrate much. लेकिन वो खुद तो aerobic है, खुद aerobic है, तो खुद को oxygen चाहिए. So it has very high metabolic rate and it has also slime layers. So it is very fascinating when you read about these facts. Huh? Then azospirillum is a chemoheterotrophic bacteria associated. See, this is associated. Associated to live within the roots of sorghum, palm millet, rice maize, wheat and sugarcane crops. Mainly the C4 crops. If you see here, rice and wheat is C3. But anyhow, in all the crops of this, it is associative. Associative matlab it stays in association. It is not purely a symbiosis, but it is association. Then frankia. Frankia is also very important. It is actinomycetes, frankia. It forms nodules in non-legumes. Woody plants. Here comes your two questions. Non-legume and woody plant and actinomycetes. 3 questions. So in this one line only you get 3 questions. Frankia is an actinomycetes. It forms nodules in non-legumes and woody plants and fixes N2 symbiotically. Frankia induce N2 fixing root nodule on more than 280 species of woody plants from 8 different families. Casuarina, a non-legume, is the most important actinorhizal genus of the tropics and subtropics. This whole slide is very, very important. See, everything that is written is question worthy. Everything that is written. Frankia induce N2 fixing root nodule on more than 280 species. Casuarina is the most important actinorhizal genus of the tropic and subtropic. The ability to form symbiotic association with Frankia enables them to thrive in N-deficient soil and makes them desirable for agroforestry. That is also very important. Then we have come to cyanobacteria. This is also known as BGA, blue-green algae. This was known like in the past days because at that time scientists did not have access to all those improved instruments. So they thought algae की तरह दिखता है तो algae ही होगा. That's how they named it blue-green algae. So cyanobacteria Anabina azole, it fixes N2 symbiotically with the azolla, a water fern. Very, very important, azolla water fern. It remains closely associated with special meristem and grow in the cavity of leaves. Again, very important, cavity of leaves of azolla. Excepting Anabina azole, no other BGA species are found in the cavities. The first account of agronomic potential of BGA in rice was presented by P.K. Day. All this I can specifically remember. All these things are in the soil biology chapter, which will be covered by Rohit in the upcoming days. Okay. National Research Center for BGA is in IRI 1988. Very good, Pawan. Nice information there. Microbial process and mechanism of P mobilization. This is also very, very important. Solubilization and mineralization of inorganic and organic P and the mobilization of P through mycorrhizal association play the major role to make the unavailable phosphorus into plant available form. Solubilization of inorganic P compounds through the production of organic and inorganic acids by altering the solubility. Mineralization of organic P compounds through the enzymatic hydrolysis by phosphatase and phytase. Please remember these two names. The two enzymes that can mineralize organic phosphorus into inorganic plant available form are phosphatase and phytase. Phosphatase and phytase, all these two are phosphorus mineralizing enzyme. All right. Immobilization of inorganic P compounds into microbial cell followed by mineralization after the death. Okay, fine. Oxidation of phosphate, fine. Reduction of phosphate. Okay, fine. Production of siderophore. Production of siderophore. Sidero is related to what? Sidero is related to what? यहाँ पे लिखा हुआ है tightly to prohibit its reaction with soluble phosphate. Mobilization of P through mycorrhizal association. Okay. Mobilization of P takes place through mycorrhizal association. Production of siderophore binds iron tightly to prohibit its reaction with soluble phosphate. ये statement का मतलब क्या है? ये statement का मतलब यही है. Production of siderophore production होगा. That siderophore will tightly bind iron in its structure. This iron will thus be unavailable. This iron will be unavailable to react with phosphorus and form insoluble iron phosphate, which is also known as stringite. So stringite formation से ये रोक रहा है because siderophore is capturing the iron directly and not letting it react with the phosphorus. Okay, that's how it is improving the phosphate availability, and this mobilization of P takes place through mycorrhizal association. I think all of you know mycorrhiza, endomycorrhiza, ectomycorrhiza, all these are again in soil biology only. We are just covering it here. These names, chemoheterotropic P solubilizing bacteria. This is a sure short question of 4 marks. All right, you have to remember all. Bacillus megaterium, Pseudomonas striata, Aspergillus avamori, Penicillium. Okay, all the names should be remembered. Fine. Dominant K mobilizer present in soil are bacteria Bacillus mucilaginensis, Bacillus megaterium again, Bacillus subtilis, fungus Aspergillus terreus, Aspergillus nicer, Aspergillus candidus, Penicillium bilai. Okay, all this you should read once. You should remember the examples because kabhi bhi aa sakta hai. Fine. Fine. We have come to mycorrhiza. Mycorrhiza we all know. Key points: symbiotic association, plant roots of higher plants and fungi, plant roots of higher plants and fungus. There are two types of mycorrhiza, ectomycorrhiza and endomycorrhiza. Worm fungi fall under entomycorrhiza. This is the most important group. Worm. Can anyone write the full form of worm? यहाँ पे तो लिखा नहीं है. Please write the full form of worm. These are obligate symbiont. This is also a key point. Obligate symbiont का मतलब है it can only survive when it is in symbiosis. Alone it cannot survive. That is the meaning of obligate symbiont. So if symbiosis is not taking place, it will not survive. वो obligate है. The relationship is obligate. Okay. With a network of hyphae in soil and extensive growth within the plant root. Worm fungi infect the plant, spread inside the root and produce highly branched hyphal structure known as vesicles and arbuscules. That is why it is called vesicular arbuscular mycorrhiza. Exactly, Pawan Shreyoshi, yes. Within the host cells, the arbuscules help in the transfer of nutrients from fungus to the root system, and the vesicles store P as phospholipid. The vesicles are like this. They will store P as phospholipid, and the arbuscle is the interaction site. Yes. The arbuscules help in the transfer of nutrients from the fungus. Arbuscules help in transfer of nutrients from the fungus to the root system. Okay. Here I will give you some small information. What is the mycorrhiza actually? ये plant root है. Plant root के साथ the fungal hyphae is in symbiosis association. This hyphae can go up to that portion which the root cannot travel, right? So this fungal hyphae of this, or the mycelium of this mycorrhiza, the nutrient which is present here, it will take this nutrient and make it available to the plant. Okay. So this network of hyphae, the network of this mycorrhiza is so fascinating. There is researches going on outside agriculture, you know. This research is going on to study the efficient network of mycorrhiza and to use use that as a blueprint for the roadways in the cities, you know, because the fungus makes this structure. ये लोग इतना अच्छा बनाता है this structure and this network. This network is so scientific and so nice. Research is going on to study this network and apply it in the roadways of the big cities, you know, because they have to make this network in such a way so that the energy consumption is minimum, but again the nutrient transfer is maximum. So the nutrient भी सबसे ज़्यादा चाहिए, but energy consumption भी सबसे कम चाहिए, because energy ज़्यादा नहीं ले सकते. In ideal world, we can put this mycelia everywhere in this soil, right? But that is not energetically, that is not economic, right? उसमें बहुत ज़्यादा energy लगेगा, उतना energy भी हमारे पास नहीं है. So it has to be a perfect balance of energy expenditure, also the nutrient mobilization. So these vesicles, they will solubilize, they will store the P as phospholipid, and these arbuscules will transfer the nutrients to the plants. Okay. Finally, we have come to the last portion of today's lecture, that is the radioisotopes. The discovery of radioactivity was by Henry Becquerel in 1896. The term radioactivity was first used by Madame Curie. ये जो दो क्वेश्चन है, यहाँ से तुमको 8 मार्क मिल गया, because everyone is confused. Everyone is confused. Discovery of radioactivity is Becquerel. Okay, that is not Madame Curie. The term radioactivity was first used by Madame Curie, that is not Becquerel, okay? So please be very thorough with these two lines, fine? Then these are the units, these units you can see. The amount of energy absorbed per kg of absorbing materials is called Gray, okay? All elements after lead are radioactive. This is also a question worthy statement. Then again, radioautograph. Radioisotopes are traced in the plant body using radioautograph. That's how we use it in the tracer techniques, right? That's what we use in the tracer techniques. Then Soddy proposed the name isotopes. Isotope. What is an isotope? इसको कैसे याद रखें? Isotope में P है. P है, P से क्या होता है? P से proton होता है. So P से proton होता है मतलब proton number is same. And what is this proton number? This proton number is nothing but it is the atomic number. So the same atomic number but differ in mass number. कैसे याद रखना है? आइसोटोप, उसमें P है, P से प्रोटॉन है, प्रोटॉन से एटॉमिक नंबर है, सो प्रोटॉन नंबर विल बी सेम, दैट इज आइसोटोप. Atomic number same but mass number different. The isotopes can be divided into two classes: naturally occurring isotope, artificially produced isotope. Naturally occurring isotope can be two types: radioactive, also called radioisotope, and non-radioactive, also called stable isotope, right? Stable isotope जो release नहीं करता. Then artificially produced isotopes. Induced radioactivity was discovered by Irene Curie Joliot and F. Joliot. Elements having atomic number greater than 92 are induced radioactive element, or also called as transuranium elements, right? The property of spontaneous emission of radiation of element in form of particles or rays is known as radioactivity. This is the best definition of radioactivity. You will find lots of definition in lots of places, but this is the best definition in a simple line. Property of spontaneous emission of radiation of element in the form of particles or rays is known as radioactivity. The first radioactive element discovered in the nature is uranium, and we are still fighting over this uranium after its discovery for so long. किसके पास यूरेनियम है, किसको यूरेनियम देना है, क्या है, everything is going on. The nucleus of a particular isotope is also referred to as nuclide, right? This is also very important. All these portions are very important. Radioactive nuclei are called as radionuclides, and the atoms having these nuclei are called radioisotopes, right? In a radioactive decay isotope that decays is often called parent isotope, और जो नया form होता है that is daughter isotope. Definitely here you can see this is from DK Das, I suppose. You can read these parts, you can take a screenshot and you can read later what are the different types of isotopes of elements. Here also another two definitions are given. Isobars are same mass number but different atomic number. Isobar, I don't think it is correct. Isotones are same number of neutrons but different mass number. That is definitely true. Acha, yes, it is isobar only. Okay, fine. Isobar is same mass, proton different. Yes, isobars are having the same mass number but different atomic number. Examples you can see. And isotones are same neutron. Again, you can see here N is there. So N means it is the same neutron number. So isotones are having same neutron number but different mass number. That is called isotone. Actually, I was confused about the isobar because isobar is also used in the pressure term, right? The line joining the equal pressure areas. The line joining the equal pressure areas, that is called isobar. No, it comes from barometric pressure bar. Then the laws of radioactivity. Radioactivity is a result of decay of nucleus. The rate of disintegration of a radioactive element follows first order kinetics. This is also a question. I have seen this question many times. From radioactivity, you can definitely expect questions. At least one, max it can go 2 to 3 to 4 also. Yes. So first order kinetics, this is a very known question. In my time, in my JRF in 2023, they have given a very difficult mathematics, very difficult problem of half-life. Okay, so they don't give calculators in exam, but they have given the calculation like it will take so much of your time. So some questions you can expect from this 2023 JRF I'm talking about. Jaake PYQ dekh lena, there is one sum with the half-life. The nucleus decay rate is independent of temperature and pressure, physical condition and environment. Of course, radioactivity is not dependent on any external condition. Radioactivity is dependent on the law of conservation of energy. Physical and chemical properties of daughter nucleus changes from its parent nucleus. In radiation emanating from radioactive substances, 3 different kinds of rays are there: alpha, beta and gamma. All of we know this. Then alpha particles are the material particles of mass equal to 4. What is alpha particle? You all know. That is helium 4e2. It is having 2 units of positive charge. Then it is effective in removing electrons from molecules close to its paths and result considerable ionization. This is important. Considerable ionization is important. Penetrating power of alpha particle is smaller than that of gamma rays. Yes. Beta particles are stream of electrons shot out from radioactive material with a very high velocity. They possess power of ionizing gases, though not as strong. So if there is the series of ionization, then alpha is highest, then beta, then gamma. Penetration is opposite, gamma, then beta, then alpha. Okay. Gamma rays are not charged particles, not deflected by, this is important, gamma rays are not charged, not deflected in electric and magnetic fields, unlike alpha and beta. They consist of electromagnetic radiation with very short wavelength. They are thus comparable to X-rays, only differing from the latter in their origin. The source of gamma ray is the atomic nucleus, while that for X-ray is the extranuclear source. This is the difference between X-ray and gamma ray. It has least ionizing power and highest penetrating power. So what I said, penetrating power idhar se idhar, ionizing power idhar se idhar. Fine. I don't think यहाँ पे और कुछ problem है, because everything is very easily understandable here. Gamma rays and X-rays में the only difference is that the source. Okay. Then measurement of radioactivity, all this you can read, but the most important is Geiger-Müller counter. Geiger-Müller counter you might have seen in films, you might have seen in horror films, in Chernobyl web series also it is there. That is a tube. In that tube, here you can see quantitative measurement of radiation is done by the counter tube having a chamber filled with gas and a central wire maintaining positive potential. So एक central wire होता है which is positive and एक tube होता है, the whole tube is negative. All right. When radiation passes through the gas and this whole part is filled with inert gas. So when the radiation enters here, it will ionize this gas. It subsequently produces a small pulse of current which is measured by the instrument. कैसे ionize करता है? The inert gas will come, that will knock out the electron. All those electrons will move towards this middle part and एक electron दूसरे electron को knock out करते करते जाएगा. It will form a electron avalanche. It is called a electron avalanche. ऐसे करने के बाद सारा इलेक्ट्रॉन मिलके यहाँ पे जाएगा. So there will be a flow of current that will further be amplified in an amplifier. और उसके बाद this current will tell that radiation का intensity कितना है. The problem with Geiger-Müller counter is It cannot tell which type of radiation is there. It cannot differentiate between alpha ray, beta ray, gamma ray, but it can tell how much radiation is there. Kitna hai, it can tell, okay? Chamber is filled with low pressure argon or helium gas. As I told, it is an inert gas. The potential difference between central electrode and the walls of the chamber is about this much, with the negatively low voltage in the ionization chamber, okay? Solid scintillation counter is also very important. All these are given very beautifully in the soil analysis book. If any one of you has that, you can read that. That is very nicely explained, all these instruments. Solid scintillation counter, in this device, radiation is absorbed and causes emission of photon of visible light. For example, if beta particles are to be studied, a zinc sulfide phosphorus can be used. The scintillating counter can efficiently detect both gamma ray and charged particles. Solid scintillation counter is important. Liquid scintillation counter is more important than solid scintillation counter, okay? So this device is generally used for soft beta emitters and alpha particles. It can also be used for hard beta by applying certain counting techniques. The recent liquid scintillation counter consists of two photomultiplier tubes. This is very important. A coincidence circuit and a pulse summation circuit. The widely used organic scintillator is PPO. Iska bhi same hai. There is photomultiplier tubes and there is a liquid in the instrument. So when the radiation enters, there is emission of light, which is multiplied by the photomultiplier tube, and that will tell how much radiation is entering or exiting the system, okay? Then mass spectrometer. Mass spectrometer is device to detect and determine stable isotope of an element. Don't worry, all this will be again covered in the instrumentation part, which will be taken by Rohit, you know, Rohit. So the mass spectrometer is widely used to determine the ratio of 14N, 15N samples, since 15N is used as a stable tracer. I was telling in the last class about the tracer technique. So this tracer technique में हमें पता कैसे चलेगा the difference between 14N and 15N, that is mass spectrometer. So 15N trace करके हमें क्या पता चलेगा? The what pathway it is taking, what biochemical pathway it is undertaking. Yes, all those. So mass spectrometer में एक बात हमेशा याद रखना, mass spectrometer work in the principle of mass by charge ratio, okay? मास स्पेक्ट्रोमीटर वर्क्स इन द प्रिंसिपल ऑफ मास बाय चार्ज रेशियो. अभी मैं इतना ही बता सकता हूँ because in the instrumentation course this will be covered in detail, so I don't want to waste time. Available nutrient concept. So this was a request from some of our students who told A value, E value, L value समझ में नहीं आता है, so we have specifically included that. A value is given by Fried and Dean in 1952. I think you might have seen this question multiple times. बहुत बार ये question आ चुका है. Who gave the A value? Okay, Fried and Dean. When a plant is confronted with two or more sources of a nutrient element, एक plant है. When it is confronted with two or more sources of nutrient element, nutrient uptake from each of these sources is proportional to amount available in each source. So यहाँ पे टू सोर्स क्या हो सकता है? एक हो सकता है सॉइल, एक हो सकता है फर्टिलाइजर. फर्टिलाइजर से प्रोपोर्शनल अवेलेबल है से 60%, सॉइल से अवेलेबल है 40%. So it is telling, so each of these sources is proportional to the amount available in each source. So plant कौन सा सोर्स से कितना न्यूट्रिएंट लेगा, that is dependent on this proportional to the amount available in each source. So ये इसका equation है, A equal to B into 1 minus Y by Y. Amount of available nutrient in soil is A, amount of applied fertilizer nutrient is B, and Y is the proportion of nutrient in plant derived and fertilizer nutrient. Okay, I think this is clear. Then next, it is E value. Of course, it is E value. E value किसने दिया था? Russell. This also I think you have found many times in the questions. This is also a PYQ. Plant available phosphorus expressed as E value is direct implication of isotopic dilution technique. E value refers to amount of phosphorus on surface of soil particles and in soil solution exchangeable with orthophosphate ions added in the solution. Okay. Refers to the amount of phosphorus on surface of soil particles and in soil solution. कितना surface में absorbed है, कितना solution में है. That is the E value. This part I have made extensively bold because I have seen this question in statement based question. The difference between E value and L value is E value does not require growing of the plant. It can be done in laboratory only. It can be done in vitro. Whereas L value, it requires the, where it is written, it requires the growing of the plant. So the difference between E value and L value is it requires growing of the plant. इसको कैसे याद रखना है? Because question will come from this only. किस में plant grow होता है, किस में plant grow नहीं होता है. So जहाँ पे L है, L comes in plant, right? So जहाँ पे L है, that comes in plant and that will require growth of the plant. E value में तो plant में कोई E नहीं है, so E will does not require the growth of plant or crops. अब L value क्या है? L value किसने दिया था वो याद रखना भी बहुत easy है, because L value was given by Larsen, L and L. Okay, so L value is the expression of labile soil phosphorus. What is this meaning of L? This L is nothing but labile soil phosphorus. It is also measured by isotopic dilution technique. Here also it is the same. So it refers to amount of phosphorus in soil and in solution that is exchangeable with orthophosphate ion. You can see both of these are almost same. The only difference is it requires growing a plant in the system. It does not require growing a plant in the system. Is it clear? Same trick you used to remember. Very nice, Pawan. That is the only trick we use. Yes. So A value, E value, L value क्या clear है? Please, if it is clear, drop a thumbs up. So I will also feel a bit good about myself. So these are our courses that you can see. L value also known as Y value. Okay. Y value के साथ Y index को ये मत कर देना भाई. Y index is used in soil. Huh? So these are our courses, as you all know. This is the target batch for 2027. You can recommend your juniors if you like, or whoever is giving in 2027, you can take this. You can see I will also be there as a mentor who will be taking this. And this foundation course is of 2 years. So whoever is in second year or third year now, Y index is given by Martil and Russell, and that is used in clay minerals. Yes, Pawan, correct. And also you can take the mock test bundle, you know. Mock test bundle, I think all of you might have taken. That is comes only for 399, and this one 2026. So I don't think this is required anymore because only few days are remaining. All right. So we have covered almost everything, but जितना ISSS में है उतना तो हमने कवर कर लिया. But what about जो ISSS में नहीं है? जो ISSS में नहीं है और लेकिन क्वेश्चन आता है, वो भी तो हमें कवर. राइट. सो जो ISSS में नहीं है वो भी तो हमें कवर करना पड़ेगा. नहीं तो तुम क्यों यहाँ पे 3 घंटा टाइम बर्बाद करके why are you seeing this, है ना? सो क्या क्या ISSS में नहीं है बट स्टिल वी विल कवर. Sir, from where I can get this PDF and previous PDFs? You can see जो डिस्क्रिप्शन बॉक्स है वहाँ पे बहुत अच्छे से लिखा हुआ है. यू कैन रीड दैट डिस्क्रिप्शन बॉक्स. देयर इज वन गूगल फॉर्म. दैट गूगल फॉर्म यू नीड टू fill up, and once you fill that up you will get the PDF instantly. And otherwise this PDF will also be uploaded in the mock test bundle also. If you are having that bundle you can get this. If not then okay. So what extra we are going to read? Is it okay? Like are you guys having energy? थोड़ा सा जो एक्स्ट्रा चीजें हैं जहां से क्वेश्चन आता है, आई वुड लाइक टू टेक दैट ओनली इफ यू टेल. अदरवाइज तो ठीक है, चलो कोई नहीं. इफ यू गाइज आर रेडी टू लिसन, इफ यू गाइज आर स्टिल विथ मी, देन ओनली आई विल प्रोसीड. बाकी ISSS का तो जितना है एवरीथिंग हैज बीन कवर्ड फ्रॉम फर्टिलिटी पोर्शन. यस सर, बता दो. वेरी गुड. सो दिस आई हैव स्पेसिफिकली प्रिपेयर्ड notes for you. Soil fertility evaluation. Soil fertility evaluation is very very important. Soil fertility evaluation का methods mainly है soil testing. We all know soil testing. So soil testing is there. Then the next part is of course plant analysis. These things all of you know. Soil testing and plant analysis में क्या अंतर है? Soil में please cover agrochemicals. अरे agrochemicals का अलग मैराथन होगा भाई. That is a different full huge topic. That will be a different marathon. Don't worry. अभी के लिए fertilizer में जो extra है, we will read that, huh? So soil testing, plant analysis, and biological test. So there are mainly 3 methods of soil fertility evaluation. ठीक है? इसमें से ये दोनों तो तुम लोगों को पता है, सॉइल टेस्टिंग में क्या होता है एंड प्लांट एनालिसिस में क्या होता है. and also this will be covered by Rohit also in the upcoming class. So this biological test is not there in ISSS, अच्छे से दिया नहीं है, but lots of questions come from this as well. Okay, so we will see the biological test. Biological test, I will write it as biotest. It can be divided in three parts: higher plants, then microbes, then okay. This one we have already seen, so this one we will not discuss. So biological test mainly done by higher plants and microbes. All of you know that. So in higher plants, these two tests are very, very, very important. These two tests are very important. I think you have seen some questions from this, or you have seen this somewhere or anywhere. देखा तो होगा तुम लोगों ने पक्का, but it is not that clearly given in ISSS as far as I remember. Right? सुना सुना लग रहा है ये दो टर्म, और it is feeling like ये क्या है, what is this, from where this is coming, कहाँ से आ रहा है, ऐसा तो नहीं है ना? I think you all know. You have seen it in T. Ramesh objective book. It is there in G. Mahajan objective book. हर book में मिलता है भाई. And in PYQs also you will find it. ठीक है? Microorganisms में क्या क्या आता है? Aspergillus niger test, then Azotobacter plaque test. ठीक है, इतना सारा I think all of you know. Aspergillus niger test, Azotobacter plaque test, then Mehlich's Cunningham plaque method, and Mulder's Aspergillus niger test. Okay. Now हम लोग बहुत ही short में in one sentences we will try to understand which one is what, which one is what. Again I am not going in the plant test and the soil test because I know all of you know that उसमें नया कुछ नहीं है, right? So coming to Mr. spot culture. Mr. pot culture test में क्या है? Here you have to remember, oats was grown as the crop. Oats was grown as the crop, and oats was grown in a pot, and it was provided with different combinations of fertilizers, N fertilizer, NP fertilizer, NPK fertilizer, and then all the yield records were tabulated and it was compared. Okay. Then in the please remember, these are soil fertility evaluation techniques, है ना? So we are evaluating soil fertility by means of seeing the plants. So this is a very easy experiment. Oats को grow करो, different type का fertilizer दो, जिस fertilizer में ज़्यादा yield आ रहा है, that is the better soil, है ना? Then Neubauer seedling method में क्या है? Neubauer seedling method was given by Neubauer, and इसमें जो है, इसमें the seedling that was used was rye. So these are the questions. Here oats and there is rye. These are the questions. Here also 100 seedlings of rye were fed exhaustively on 100 gram soil mixed with 50 gram sand and different nutrients. The total P2O5 and K2O uptake was calculated and the blank value was deducted to obtain the root soluble P2O5 and K2O. So in this all N, P and K was considered, but in this only P and K was considered. A question कैसे आता है? Which crop is grown in Neubauer seedling method? Which crop is grown in Mr. pot method? And then for which nutrient this test is done? For Neubauer seedling it is P and K. For Mr. it is N, P and K. इतना सा ही question आता है. Fine. इसके बाद जो है that is the measure. इसके बाद क्या आता है? This part. This is the most important part, this using microbes. यहाँ से question I have also covered in the most expected series. If you have seen that, you might remember, है ना? So यहाँ पे कौन सा microbe use होता है and कौन सा nutrient के लिए use होता है, that is very important. Okay, so first we will see Aspergillus niger. The Aspergillus niger test, it was given by Mihalik, and it is only for all right. Is it clear? भाई कुछ thumbs up कुछ डालो यार, because otherwise I am feeling like I am only writing alone and nothing is happening. Then after Aspergillus it is it is Azotobacter plaque test, है ना? It is Azotobacter plaque test. It was given by whom? Can anyone tell me? This is also called another technique. This question I have covered in the most expected series also. This is called Azotobacter plaque test. This was given by who? This is very, 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 very important. Okay. All right. Saket and Stewart technique. This was given in 1931. So match the following में आ सकता है. Azotobacter plaque test was given by who? ठीक है. Saket and Stewart technique में which microorganism is used? That is Azotobacter. Okay. And इसके बाद which is the nutrient? Azotobacter has the special ability of taking up this in large quantity, and they have special behavior in the deficiency of P, K and Ca. So majorly it is done for K and P. See? So we use compounds like potassium sulfate, sodium phosphate and potassium phosphate as well. Azotobacter culture incubated for 72 hours, and so यहाँ से याद रखने का यही तीन है. Azotobacter plaque test, who gave name of the scientist and what are the nutrients that can be estimated through this. Okay, then A value technique तो हमने बात कर लिया, and then what is remaining? In this also specifically used for, if you have notebooks and all, please note this down. This is very important, or you can run this video later and see whenever you want to read. So Mihalik's Cunningham-Ella plaque method, the organism Cunningham-Ella is sensitive to the phosphorus status of the growing medium, and this normally Cunningham-Ella oligans is used. In calcareous soil, Cunningham-Ella black clay sana is used. These are not important, but this is specifically for P deficiency. So इसमें क्या याद रखना है? Mihalik याद रखना है, Cunningham-Ella याद रखना है, and P याद रखना है. ठीक है? So everything is done. Finally, only we are in the, सिर्फ एक बचा है, which I wrote there, that is Maldar. Maldar का भी सेम है. Maldar का Aspergillus test. Okay, Maldar's Aspergillus test. It is for the deficiency of Cu and Mg. Yes, Pawan, you were थोड़ा सा आगे चला गया था. So Maldar Aspergillus niger test. This is used for delineating the deficiency of Cu and Mg. This method can be also used for molybdenum, cobalt, manganese, sulfur and zinc. So ये सारा लिख लेना भाई. This is extremely important. This is very important. That's why मैं अलग से इसको cover किया. कहीं पे कुछ जगह अगर छूट गया तो you tell me, I will go to that slide. but this is very important. So this is what? This is fertility evaluation. Fertility evaluation of soil. Soil कितना fertile है वो हमें soil test से पता चल सकता है, plant analysis, plant tissue analysis से पता चल सकता है, and we have this specific biological test जिससे हमें पता चल सकता है. So जो specific biological test है उसमें higher plants से जो test है that is Mr. spot culture and Neubauer seedling method. Then microbes से जो test है that is Aspergillus niger, Azotobacter plaque, Mehelix cunninghamella and Mulder's Aspergillus. Okay. Then third is A, E and L value which we have already studied. Then Mr. में क्या याद रखना है? Mr. में याद रखना है oats used for N, P and K. Neubauer में rye used for P and K. Then this is the most important part. This is the microbes. Aspergillus niger, Mehelich available K2O use करता है. Azotobacter plaque test given by Sackett and Stewart. This question I have seen myself in PYQs. Who gave the Azotobacter plaque method? As Sackett and Stewart in 1931, and it is specifically for P and K. Then Mehelich cunninghamella, it is specifically for P. Specifically for P, यहाँ पे Mehelich याद रहना चाहिए, cunninghamella याद रहना चाहिए. And then Mulder's Aspergillus test is for copper and magnesium. It can also be used for MO, CO, MN, S and ZN. So that marks the end of the marathon. I think all of you have enjoyed it. Jitna bhi tha, it was very good. And last class and this class, I think that is more than enough for soil fertility. Isse zyada soil fertility mein question nahi aayega. Okay. So there is another thing that I would like to tell you. Okay, if you guys want basic chemistry and biomolecule marathon, you have to let us know in the comment section. Till now, we have only received 3 comments in the videos. Sirf 3 requests say we cannot conduct a marathon for 3 people. At least we need 10 to 15 comments asking for the basic chemistry and biomolecules. So if you think, only if you think basic chemistry and biomolecules marathon is required, I will take that marathon, but I have to prepare and it takes a lot of effort in preparing these slides and reading. मुझे भी पढ़ना पड़ता है ना भाई. I also have to read basic chemistry. I do not need it anymore. See, I am in PhD. I do not need it anymore. Only for you guys. If you need it, we will be conducting it. So you have to comment it, not in the live chat, in the comment section of the video. When this video will be uploaded, in the comment section of that, you please, if you want, you can ask for that marathon. The management will see that and then we will start preparing for that. Basic chemistry and biomolecule. That is not under our consideration till now. We have only received 3 requests. At least we need 10 to 15. Okay. Second part is after this instrumentation, miscellaneous topics, biology and all marathon will be covered by Rohit. And finally, in the last day, I will come again with agrochemicals marathon. Agrochemicals is a tough part, I know. That is also very much required for JRF as well as SRF also. In SRF, part B mainly consists of agrochemicals, water science and agronomy. So that marathon will be done. But basic chemistry and biomolecule, if you need, please comment in the comment section of the video so that the management sees it and we will go on with that. Today's session, if you have liked today's session, please let me know in the live chat or please let me know in the comment so that I will get motivated and I will work further hard for bringing these sessions for you. And also today there was this course launch video of AgriAddict that has been already uploaded in our YouTube channel. In the video section you can see. So I would request you to see that video, see who are the mentors for the next year, and you can share that to your juniors. And also you can drop a comment there also if you like. So if you would have liked the session, please let me know and go on. Fertility, I think this much is enough. And also from new ISSS and from treatise, please read the sections of soil health card. Soil health card is very important, which I have not covered here, but you have to read soil health card. Then please read the, what is that? PUSA STFR method. PUSA STFR method, the soil test fertilizer recommendation, that one. उसका कितना parameters है, कितना parameter cover कर सकता है. Soil health card, when was it started, which year, which month, how many parameters it can suggest, till now how many soil health cards have been distributed and what is the purpose of soil health card, how this soil health card can be further improved. All these things are in that. In the new ISSS, that chapter soil testing and fertilizer recommendation, that is too good. If possible, please read that chapter. अगर तुम लोगों के पास PDF नहीं है, you can contact me, you can contact AgriAddict, we will provide the PDF also. Please read that portion, that is very, very important. बाकी fertility evaluation से, this fertility evaluation was important, which I have covered. बाकी मुझे नहीं लगता यहाँ से और कुछ आ सकता है. In the instrumentation and the analysis class, Rohit will cover the testings, nitrogen and phosphorus and all those tests. So those will be covered in that. Baki, I think it's good. So if you have liked the session, please let me know, and I will be ending the session because yes, I think we are done. Okay, Pawan, thank you. I don't think anyone else is there, but if you are there, and also again, I will say the same thing. If you want basic chemistry and biomolecule marathon, let us know in the comment. Then only we will be able to prepare it and we will go forward with it. Otherwise, we will have to drop that plan. If you think it is enough, you can read yourself. That is very nice. We will also appreciate that. All right. So thank you everyone. Thank you Pawan. Thank you Shreyoshi. Thank you all other who have joined. It was a great interaction with you people. And abhi mera class और कुछ दिन बाद आएगा. So I will not be meeting you guys for quite a period of time. So till then, please stay safe, healthy. अभी कुछ दिन बचा है. That is very crucial for JRF. Yes. Give your full. अभी lapse नहीं होना चाहिए. Give your full preparation and all the best. All right. Good night everyone.